All right. Wait till Mike gets there. We don't want to throw a mic at them and see if they catch it.
I don't. You have plenty of room. You can have some of my chair.
If the mic doesn't hear it, no one out there.
You're a lady like that. He's gonna bring you a chair over there.
Yeah. My wife's out there, so I gotta make sure. Yeah, definitely. Yeah.
John, so now we're gonna need a chair on that end, it sounds like.
You can scoot here.
Where would you like this chair?
Somewhere, I can be at the end. Good morning, everyone. I'm Sarva Chennarajaras, co-founder and president of Mithil Prasad Foundation, and also DARPA Member Engagement Coordinator. I wanted to start today by sharing a personal story with you. So it was July 4, 2016. We wanted to go see fireworks because that was one of Mithil's favorite thing to do. And we usually used to go to a hilltop on our neighborhood because we used to get a nice panoramic view there. But that day, Mithil didn't want to get down from the car. He insisted he wanted to stay back. But we were like, he had a little bit of walking problem during that stage of his treatment. We wanted him to take his mind off of it, and we said, why don't you come along? But he insisted that he wanted to stay in the car, and he wanted me to stay with him. So when my husband and daughter went off to see the fireworks, he immediately turned to me and asked, I want to know, what are the statistics? What chances do I have with this tumor? That took me by shock. I didn't know how to answer this. So I just told him, hey, numbers are numbers. Everybody is different. It doesn't tell the whole story. He knew I was not telling the truth, and I hope he understood why. Then he looked with his beautiful big eyes. We hugged and cried for the rest of the time. I wish I could say to him, Mithil, you have TIPG, but it's okay, we'll be fine. But for that to happen, we need to find new ways to treat this tumor. And one of the things that has made leaps and bounds advances is in data analytics and AI in healthcare. And that's what our session is today about, to figuring out the power that patients' data holds in forging ahead in new discoveries, new therapies. So basically, we have a panel of researchers and advocates who are going to share with you the pivotal role that patients and their data play in this new, finding novel treatments. I just want to remind, we'll have a question and answer session at the end after all of them present, just so that we give them enough time to cover their material. And then please note down your questions as the sessions go, and you'll have time to ask them later. Now to our wonderful presenters. I apologize for the abbreviated version of your introductions. Their full bio is on the speaker section of the Brainstorm Summit website. I know these are all highly accomplished individuals, so I feel bad, but just to keep the time on track, I want to just give an abbreviated version.
There are lots of seats in the front. It's like church. You just come late, you get in the front row.
Olena Vasque founded the Tree House Childhood Cancer Initiative in 2015 at UC Santa Cruz. She holds a PhD in bioinformatics from the University of British Columbia. Ripley Martin is a patient advocate, board member of Carson Leslie Foundation. His son Cooper is a 5-year-old medulloblastoma survivor. Ashley Margol is the director of clinical trial development and co-leads the ATRT working group at PNOC. She has designed a comprehensive ATRT registry and is co-chairing an innovative clinical trial for children with recurrent progressive ATRT. Thank you all for doing this. So, Lena, to you.
Thank you so much for having me. It's truly an honor and privilege to be here. And so I'm going to speak today about clinical utility of shared data sets. So I'm from UC Santa Cruz, and at UC Santa Cruz we don't have a medical school, but what am I doing here? Our expertise is in genomics and in large data sets, and so as Sarva mentioned, in 2015 we decided to make a difference in pediatric cancer, and we started this program called Treehouse Childhood Cancer Initiative to see how can we leverage our expertise and large genomic data sets to advance pediatric cancer research and to identify new therapies for pediatric cancers. And I'll tell you a little bit about what we do exactly. So I'm not going to go through the slide because you've heard at this meeting repeatedly that pediatric cancers have fewer mutations than adult cancers, and they're often driven by epigenetics, which means that we need to look beyond DNA to identify novel treatment targets. And so in our group, we look at RNA, which is this molecule that describes which genes are turned on and off in the tumor. And we hypothesized when we started this that by doing these RNA sequencing comparisons, and I'm going to tell you what they are in a moment, we can identify additional genes and additional proteins that we can target by identifying genes that are overexpressed. So that means there is too much RNA that's expressed, and identifying the whole pathways that are overexpressed. And so that will help us identify additional avenues for targeting these tumors because there is not that many mutations. And so why big data and why data sharing? Well, it's because RNA expression measurements are relative. So here is an analogy I have. If I show you this one dog in isolation, you have no idea whether it's a big dog or a small dog, right? So you only know that this is a big dog when you put him next to other dogs, and so now it's evident that this is a big dog. And so we use this analogy for studying RNA. So when we look at one tumor and look at RNA levels of that tumor, we have no way of saying what's too high or too low. We need to look in the context of other data sets to say, okay, this gene is really unusually high in this tumor. And so because of that, we spend a lot of time building these large RNA data sets. And so we have data from various initiatives, including CBT and PNOC that you've heard at this meeting, Target, St. Jude, Nationwide Children's Hospital, so Elaine Mardis's data. And so we're building these compendia that we can use to compare individual patients to try to understand the disease and identify novel treatment targets. And this is a visualization we call the tumor map, where every dot is a tumor RNA profile, and then we can lay out this map and look at the similarities across different tumors and color them by disease. We can color by age and various other characteristics. So what's very important is our data are uniformly processed so that we can compare data to each other across different institutions, reduce batch effects. We often face this barrier where raw sequencing data cannot be shared for regulatory reasons. And so we The way we address this is we bring our computational tools to those institutions, and then they process the data there and then share the output with us. And so that's how we get around some regulatory issues. You may hear from the rest of the panel about kind of the difficulties of sharing information. So I'll share with you a couple of cases of how this has been useful in the clinic. So this is first case is a young adult with Leishmania-associated glioblastoma. So Leishmania is a disorder that is associated with inherited TP53 mutation, and so these individuals are predisposed to a lot of cancers, including brain cancers. And so this particular patient had a lot of different treatments. By the time we saw him, had 6 different surgeries. And so we analyzed RNA profiles from this patient. Sorry. And so here is the design of what we do. So we take a tumor, and then that tumor is subjected to RNA sequencing, and then that RNA data is mapped to our tumor map that I showed you to see what tumors is this patient's tumor is most like. And then we do the analysis to look for these genes, overexpressed genes. So RNAs that are too high in the tumor or too low compared to other cancers, and then which of those could be targeted by drugs. And so in this particular case, I don't know, sorry, I don't know why it's cut off. So what this slide is meant to show is that this particular tumor was unique in that it was similar to kind of unexpected brain tumors, which we can only see when we compare to this large compendium. And then we were able to identify, so this is a picture of a pathway that we identified in this tumor. So all these genes that are color-coded are highly expressed in this patient compared to our compendium. And one particular molecule there, STAT, could be targeted by therapies, and which was done in this case, and stable disease was achieved for a month, which does not sound long, but actually this patient was not responding to any therapies for that long, and so this was success. And I know I'm running a little. So another case here. So this is a 13-year-old boy with recurrent parasitic astrocytoma that had this BRAF fusion that Elaine Martis talked about yesterday, and so this tumor was also not responding to a lot of the therapies that were given. And so RNA sequencing was analyzed by Treehouse, and so again we mapped this tumor to the map, showed that this is similar to other BRAF fusion cancers, and then we were able to identify a pathway that was overexpressed, suggesting some targeted therapies. In this case, it was proteasome inhibitors. And then actually cells from this patient were tested, and it was shown that yes, indeed, this tumor was responding to proteasome inhibitors. And so I think what I was trying to show to you is that comparing individual patients' data to these compendia, we call them compendia, aggregations of other cancer data from other cancers is meaningful and can provide clinical insights. And so for us to make progress in genomics, we really need to share the data. And I've shown you examples with RNA. The same goes for methylation, for imaging, all kinds of data. It's only through sharing so that we can compare each case to this agglomeration of data that we can make progress and derive these new insights. And just very briefly, I just wanted to say that we have, so we piloted this genomics education program we call 2B Treehouse UCSC Bioinformatics Immersion. And this provides an entry into genomics for people with no genomics experience. So we started it for our UCSC students because we wanted people to get passionate about pediatric cancer genomics specifically, because we need more brains to work on these diseases. But recently we've realized that patient advocates are another group who could benefit from this. So either to learn more about genomics and to understand their tumor profiling that was done on their child's tumor or their family member's tumor, or to help get into research. So if you're interested in this, we're in the planning stages. Please talk to me and my colleague here, Holly Beal, who actually created the 2B program. We'd love to hear from you. Okay, thank you so much, and I'm sorry if I was...
I just wanted to take a couple of seconds before I hand it to Ripley. I realized I missed Lisa Ward, introducing her, but then I thought she doesn't need any introduction. But I did want to mention one thing. Misha, myself, and several others have been working with Lisa for a long time now. One thing I can tell about Lisa, she's been the champion behind bringing the DIPG-DMG community together, especially on the data sharing front. So, we owe a lot to her and the work she's been doing with everybody in the community to really bring this, front and center. So.
So the story you just heard was taking a lot of little stories and turning them into a big story about one question or one specific type of question. And the only way you can make any big data question like that work is if you start at the really small level at an individual patient journey, and how that patient journey can be curated in such a way to handle your own patient's care, and then learn from both your patient and all the other patients that look like your patient to add them up into something that can support research. And that research might exist in academia, and it might exist on the corporate side. And they're actually slightly different. So to start, it's helpful to start with an individual patient journey. And this is my son, Cooper, before he was diagnosed, during, and then post-diagnosis. So Cooper was diagnosed just after he turned 6 years old. He was a medulloblastoma diagnosis. He was also a speaker at the Golden Toast a couple of years ago. And yesterday he ran in his second cross-country meet, which is fantastic. When people ask me how Cooper's doing, that's like an incredibly relative statement. So there's the kids who are running like a 6-minute mile and their parents are very excited about what that looks like. Cooper's pretty atypical for a 12-year-old. He talks to his endocrinologist. He's had 2 cataract surgeries. He has tons of things going on at school that are not normal for a 12-year-old, but I will take that every single day. In order to make Cooper understandable to his clinical team, he's actually looked at totally differently. They don't necessarily look at his picture, and I think some of the people may actually don't want to look at his picture. They want to look at him this way. They want to say, oh, he was diagnosed with dizziness and balance issues that we figured out was hydrocephalus that was going on because he had a tumor in his 4th ventricle. He was actually initially diagnosed, not diagnosed, but our ophthalmologist said you need to go get an MRI. And then we sent him into Boston Children's. We figured out there was a mass, but we actually didn't know what to call it yet. No one really does. They just say, hey, you have a mass. But getting to a point of definitive diagnosis requires total resection. And then being able to get into your cell and molecular pathology to say, hey, we've taken your tumor, we've looked at it under a microscope, we've run sequencing on it, we've figured out that there's no tumor in your spinal fluid, and we're getting closer and closer to what we actually want to go do before we even pick our treatment selection. So we also figured out that he didn't have some of the amplifications that I think briefly were mentioned earlier. So he had no MYC, he had no P53 amplification, but he had some anaplastic cells that were making the clinicians a little bit nervous. So all of this is just to get to the point where they start doing some actual treatments beyond just resection. So Cooper had an interesting journey in that the Boston area medical complex is rife with politics and fiefdoms. So we had our initial diagnosis and surgery at Boston Children's, which is affiliated with Dana-Farber, where he had his MRI. He happened to have surgery in an imaging-enabled operating room, of which there are a very small number in the country. So he had intraoperative MRI, he had intraoperative ultrasound that allowed for resection that doesn't happen in ways that if you're in a more remote location and you have emergent surgery to remove a medullo, you're going to have a general surgeon who's on call at 2 o'clock in the morning and do the best they can to debulk your tumor, which is totally different from somebody who's done a whole bunch of these things with really great cameras in the operating room that can help them figure out how to remove tissue. We also happened to go to get our protons at Mass General, not at another facility. Mass General and Boston Children's sort of share data, but they also have their own research objectives and they're part of different networks. The The chemotherapies that we gave him are essentially antique drugs. I'm sure many of the people in the room know this. We gave him mustard gas and platinums and all sorts of things. And in the conversation, if you were in the keynote earlier, most of those actually don't cross the blood-brain barrier, and they sort of kind of help, and they're marginally additive to surgery and radiation, but they really don't know what else to give you. But because most medullos are generally survivable, I'll paraphrase what doctors say, let's start here and see if this works, and if this doesn't work, then we'll go figure something else out. That's very different from other tumor types where you know you're not gonna have a course of therapy that will work, so you pick other more aggressive trials very, very early in the process. So our tumor map really turned into this. We're kind of group 3, maybe group 4, but basically medullo is not very well designed in terms of how to de-average group 3 and group 4 medulloblastoma. And all of our choices led into, hey, you're one of these categories. We're trying to figure out what this looks like, but we don't really have a clear picture of how to create a different therapy pathway for you, aside from we're going to start with standard risk and we're going to go from there. I'll kind of pass through these real quick. So Cooper's also had this other set of doctors. Because he's a survivor, he has a series of other specialties that are now part of his care. He has a huge ophthalmology record, an audiology record, endocrine, urology, neuropsych, and then a range of other genetically informed screenings that we're doing on the imaging side to make sure he doesn't have recurrence or secondary tumors. There's also a significant amount of social determinant data and nutrition and exercise data, and this is all just for one kid. So if you want to start asking any kind of big clinical question, this obviously requires something like a registry that you'll hear about in a moment to aggregate all the patients who look like Cooper across anything that might look like this type of disease. Now, the way this stuff happens in the past is there's like a medullo registry or a high-risk medullo registry or an ATRT registry or a DAPG registry. And sometimes they're actually similar and say, hey, maybe that data and that data need to look the same. But the way that this has happened in the past is like, think of everything as a pipeline where you need to take the kids, groom the data, get it exactly to answer the kind of questions you want, and then the academic researchers can say, okay, data's clean, now I can go ask my questions. That's like 80% of the work is just getting the data into a place where you can ask a question. It's a total pain in the neck. Now, we live in 2023 when like LinkedIn can tell you friend of a friend of a friend, no problem. And the way that that is architected, just the way that the cloud data sets are architected to support, they call it graph data models in terms of relationship maps, the kinds of relationship maps you were showing earlier, allow you to create inferences across lots of things that may not appear to be the same, but are actually similar. And the way that a personal health record, a longitudinal personal health record that covers all these things is set up for one disease, if you do that to do one disease and then a totally separate stovepipe for a different disease, you are making it so architecturally you cannot find inferences across those things, which is a total mess, especially when you're dealing with very small sizes of individual kids who have some of these mutations. So there's this network. So she did a network map of drugs. I did a network map of all the people working on this stuff. And there's a huge map of 37 consortia that say that they're working on pediatric neuro-oncology, but none of them actually have a shared data model to describe any of this stuff. That's a little bit of a shame statement for people out there. Now, if you take some of those concepts about like what modern cloud and modern SaaS look like, and you say, oh, my individual patient records, so what they're doing with the DIPG navigator, or what you're doing with ATRT, or what Medullo might do, and say, hey, we could actually look at this, and the technical term is a multi-tenant way, where you have all these data that exist in an environment, and how I see my data is slightly different from your data, but it's all basically in the same place. It allows you to do AI, and it allows you to do inference mapping across different sources that otherwise take way too much time and energy to go build. So you can think about it in terms of like the number of bake sales that are necessary. Imagine if you had to do 10 bake sales or 10 10Ks, and you knew that the first 8 of those were to do stuff that was the exact same from a plumbing standpoint across every single disease. So all of the overhead that's necessary to support all this work is almost shameful that that needs to exist out in the world, because if you say a project's going to cost $1 million, 800 grand of that is the exact same cost that needs to exist across every single question that's asked. So those are my parting thoughts in terms of like, all of Cooper's data doesn't exist in one place. Our treating institutions hold samples and they try and stovepipe data. His surveillance protocol is genetically informed. as a survivor, his personal health record and registry should actually all be the same thing, not viewed as these separate pieces of information that go somewhere. And it needs to be bidirectional. We need to understand what's going on with our research and vice versa. And I will close there and hand it off to the registry folks. Thank you.
Good morning. It's a pleasure to be with you today. So I am not going to fix the problem laid out by Ripley, but I'm going to take us a few steps further and pretend kind of in an ideal world, if we were able to have such a system where we had all this data collected in one place, what we could eventually do with that, and how we're trying to come up with a proof of principle. Okay. So most of you are very familiar with randomized controlled trials, but just as a quick review, in a randomized controlled trial, you enroll a large number of patients. They are randomly assigned to what we consider a control arm, which in some cases is a placebo. In other cases, it is what's considered standard of care therapy at that time. And the other arm actually gets the intervention that you're trying to investigate. So in this example, they use, here's a group of people who are looking for work. Some of them get an intervention like job training, and at the end of the day, you compare the number of people who successfully got a job to those who didn't between the two groups. And this allows you to define the effect of your intervention. So this is a way of proving causality. We can prove that this intervention worked or it didn't work. And there are a lot of advantages to this. So the biggest advantage is that you're confident in your results. So you can say this wasn't due to bias. Again, we can show causation. And that's why these trials are really considered the gold standard when you talk about regulatory agencies. That being said, there are a lot of disadvantages. And the biggest disadvantage is really just around sample size. So it takes a lot of patients to conduct a randomized controlled trial. And if you're looking for an effect that's moderate, or you're looking for a change that's not that big, so we want to increase survival by 10%, the smaller the effect size that you're trying to find, the larger the sample size needs to be, the more number of patients you need to enroll. And then obviously from the parent side, you've got a number of subjects or patients who are being enrolled in a control arm. Fortunately, in pediatric studies, that's not usually a placebo, but it's still a standard of care that clearly isn't good enough as we're trying to improve upon that. And then given the large sample size, obviously these trials can take a really long time to run, sometimes upwards of 10 years. And really, by that time, the risk of the therapy could be outdated, the biology that drove your initial study design could be outdated. And so this can be really, really challenging when you're considering rare diseases that already have poor outcomes. So then that begs the question, can we do this differently? And this conversation is being had in a number of circles at this time. And it's really around how can we design clinical trials better, especially for rare diseases. If you're talking about other types of cancer where you've got large numbers of patients, sure, randomized controlled trials make sense. But we don't have that luxury in pediatric oncology, and especially not in pediatric neuro-oncology. So what are some other options that we have? And one of those options is using what we call external control cohorts. So I'm going to try to walk you through this. So essentially, you can think about this as an external group of patients that are going to serve as your control arm. So you open your study, you enroll subjects. All of the subjects enrolled on the trial get the intervention that you are investigating. So if we're looking at drug X, you enroll your patients, they all get drug X. And then we go find subjects that match them. So you have to have obviously a set of data that you would go to and say, okay, here's patient one that enrolled, let me go find a patient that matches this patient in this external data set. So we call this an external control arm. Interestingly, based on kind of how these trials are designed and the statistics required, you can see here that the external control data set is larger than the number of enrolled patients. So in a randomized controlled trial where you might do a 1-to-1 randomization, and so you might need 20 patients, 10 and 10, let's say. In this case, you would, let's say, enroll 10 patients, and you might need 40 that matched in the external control data set to kind of manage things like bias and other situations like that from a statistical perspective. But again, these are patients that were already treated, right? So you're not having to enroll them. So it really decreases the sample size for the trial. So there are a lot of advantages to this. They can be conducted much more efficiently. So anytime that you decrease your sample size, your trial is going to be more efficient. You avoid randomization to a potentially ineffective control arm, right? Nobody wants their child to be randomized to a therapy that we know doesn't work well. Because these trials take less time and enroll less patients, they're less expensive, and we can get to an answer faster. That being said, there are obvious disadvantages. One of those is that you might miss a small effect size. So these trials are not good at finding a small benefit. They're only going to find kind of a larger improvement in the survival or whatever your endpoint is that you're looking at. They're inherently a risk for bias, and that's just because they're not randomized. And anytime you do a non-randomized trial, you run the risk of introducing bias. And when you talk about regulatory agencies, a lot of times just a single-arm study using an external control cohort might not be enough. And so a lot of times when we've been in discussions with regulatory agencies, specifically FDA, they would really like to see kind of complementary data. So for example, if you've got really strong preclinical data that supports this, if the study has been done successfully in, let's say, an adult population that has a similar target, those are the kinds of additional data that can really help support the use of this trial. So where do we get these data? Where do we get this data to use as an external control cohort? And there's kind of two options. One is clinical trial data, which we know are sparse in pediatric neuro-oncology scenarios, and the other is real-world data. So these are data that exist out there, so patients' data that can come from the EHR or registries, which is what we're focusing on. And so you have to be really intentional when you're creating these external control cohorts. So you have to look really closely at where are the data coming from, what are your data sources, do those data sources have the key variables that you're going to need to look at and to appropriately match your patients. Then you collect this data, this is the raw real-world data, then you have to clean it, so you have to look at it, make sure that patient by patient, this makes sense. Is this patient's date of diagnosis, before their date of starting therapy, things like that. And you also have to obviously make sure that the data records are complete. You mentioned common data models, so we have to make sure that we're using data variables that make sense across different platforms. So what does date of diagnosis mean? Is that date of MRI? Is that date of surgery? What do things like that mean, and how are those relative to other studies? And then we combine the data set and we'll say, okay, for this particular patient population that meets all these criteria, here is, let's say, the 3-year progression-free survival, and that's the number that we're going to use when we're comparing to our interventional group. And kind of once you've done all of this, then you have your fit for use real-world data set. So my area of research is ATRT. So for those of you who aren't familiar with this tumor, it's rare. There's about 75 kids in the US diagnosed every year, and it's typically a disease that occurs in infants. There are 3 molecular subtypes that we have currently defined using methylation profiling, and it's very heterogeneous from a molecular standpoint. And outcomes remain poor, about 50% 3-year event-free survival, and there's really no effective cure for patients with relapsed disease. So we set out to say, okay, can we use ATRT as a pilot or a proof of principle? If we are able to create a real-world data set, can we then use this to create external control cohorts, run clinical trials that could then possibly be acceptable to regulatory agencies? So this is a lofty goal, and we're at the beginning of our journey. So kind of here's where we are. So why did we do this? Again, ATRT is rare and heterogeneous. It's only been included in about 3 prospective clinical trials, the largest, which had 65 patients. I mentioned standard of care therapies still have poor outcomes, and we still don't have prognostic clinical variables, molecular features. We don't have predictive biomarkers. We're still in the dark about a lot of things about this disease. And then, when we kind of started on this journey, we looked around and said, well, what data sets can we start with? And there are a lot of data sets out there, but again, kind of holding it to that really high standard that I showed you about the real world data, just none of them really met all of the criteria that we needed to move forward. And so we decided to, in some ways, start from scratch. So here's where we are. PNOC has created kind of the framework for us to do this. And so I lead the ATRT registry through PNOC in very close collaboration with CBTN. And kind of the big picture here is that we're collecting patients from data from as many patients with ATRT as we possibly can. So patients that were previously treated, whether they died of their disease or they're still alive, patients who have been newly diagnosed and undergoing therapy, patients who have relapsed disease. And from those patients, we're collecting as much data as we possibly can. Again, so clinical data collection, we have really, really detailed CRFs to kind of be able to follow a patient through their entire journey and gather all of the information along the way. We're collecting imaging. So I know we heard about difficulties with collecting imaging yesterday, but we're really trying to get all of the imaging from the patient into a central place. We're collecting biospecimens, so plasma, CSF, tissue as well to do molecular subtyping, and then there'll be quality of life assessments for eligible patients. Those data will all be stored. We'll have them what we call patient level, so we can track a patient all the way along their course, matching up their tumor tissue, their imaging, their clinical data, and then those will be publicly shared. One piece of this, which we're really excited about, is what we call, it's an optional tumor board. So for patients who've had relapse disease, a lot of times they don't know what to do, they don't know where to go, they end up reaching out to several of us, and it's usually the same several of us. And we really wanted to kind of honor our parent advocates and what they were telling us, and so we've given them the option to have an expert tumor board where we will collectively, along with their treating physician, review their imaging and clinical history, and then report back kind of what our recommendations are. So I think the most important thing, this is my final slide, is that collaboration is key. So as we embark on this journey, it's really going to be a collaborative effort, or else it's going to fail. And the reason that we feel like we're really going to be able to be successful is that we have an incredibly engaged parent community. We have a specific advisory board just for the registry. We've met with FDA colleagues a couple of times to get their input on the data elements. Again, we've created this really detailed database using common data dictionaries whenever possible. And then we've been discussing with other partners, right? So the CCDI and industry partners to kind of be sure that we're on the right track. And if we get to the end of the line, this is going to be something that's useful. And then we're also working, like I said, obviously really closely hand in hand with CBTN and trying to augment the cohort that already exists there by going back and asking more questions and getting more information so that that cohort can be leveraged for this project as well. And obviously, thanks to many people, many organizations who have been supportive of this, both conceptually and financially.
Here, if that's okay, because I really want to open up a conversation. I love this, and I love what we're doing in the DIPG world now too. You saw yesterday the tumor board and the collection of data, and as we became closer and closer as parent advocates to what's going on, we just had some questions. And I will tell you, the last time I took a science class was in 1988, so I don't have the answers. You're going to have to look to somebody else on the panel and those within the room. But I do, in my day job, solve problems. And you have to ask the questions to solve the problems. And one thing that I had a question about as Jace went through his time was, who's learning from his experience? Who is learning from it? He went from hospital to hospital to hospital because we live in middle America, but who is learning? And who is learning from the little girl in Wichita who never left our state? And where is that data getting collected? And is it in the right way? And how are we talking to each other? And I'm going to tell you, 2 years later, I still have lots of questions because there are places I thought had shared Jace's data who can't tell me whether it's been shared or not. And that bothers me. And it's not because they're doing anything wrong. It's because their model was set up differently. It was set up for the science of it, right? It's because they're collecting maybe 30 or 40 data points instead of having the storage for all the clinical records. But what if next year we learn there's 5 other data points we should have collected? Who goes back and finds all those records? So it just seems to me we, as parents, are able to say, hey, while we have the opportunity, let's make sure as much of the data as possible gets put in a place that we can still get to it. So when they turn off my chart, if our child doesn't make it, our child's story is not over. We can still lend that. But I have to ask some questions of those of you in the audience, and I think it is different for different tumor types, okay? And I'll tell you why. If I had breast cancer, I might not want to share everything about my experience because I might have to go back into the workforce. Somehow I might wonder if it would keep me from a new job, anything like that, right? Like my story would look different. But if I have DIPG, what am I protecting, right? What am I protecting? Don't I want, and this is what my son would say, I don't care who has my data if they can make a difference. So by a show of hands, I want to ask you, how many in this audience who have had an experience with childhood cancer would not want your data shared if given the opportunity? So how many of you do not want to share data? I'm not going to shame anybody. I'm not going to call you by name. How many just said, I do not want to share my child's data? Okay, that's fair. That's fair. How many of you would say, I only want to share it with people that work for research, academic institutions, not companies? A company that might be working on a drug to help DIPG. Researchers, like industry researchers, right? How many of you? Yeah, because the corporate might directly make money, right? Okay. Okay. How many of you feel, I just want to get it in the hands of somebody who can make a difference for the next kid with this tumor? Okay. That's how I feel too. And so one situation we have is sometimes our academic institutions, as I've been explained, and Ashley, you can tell me where I go wrong, because I don't want to go real rogue, but sometimes I understand they have policies. So it's not Ashley that wants to keep the data away, but there's policies that say you can't sell our data, you can't monetize our data. That sounds dirty, right? That sounds dirty. But the industry don't have a way to get the data without licensing it really either. So we have this whole disconnect where we have people that are in the industry side that think they have opportunities but can't get the data well. And there's not a way for the people in the academic institutions to really do it unless they're partnering on a trial, et cetera. So as patients, if we feel like, you know what, I don't care. I lost my kid's in the fight, my kid's a survivor, but it could relapse. I want anybody who can use that data to have it, then we got to make some noise, okay? We got to step up and make some noise. And the way we're doing it in the DIPG community is we're not saying there's a right way and a wrong way. We're saying you want to share your data and make sure it can get into industry's hands, you're going to do it with XCures, because X-Inform, right now, you can go to your phone, go to DIPG1 link and say, share my data. If you've had a kid in the fight in the last 5 years, if you have a kid in the fight now, share my data. They go collect it, grab it, and use it the best they can. Here's what we got to do. We got to make sure that we collect it and use it in forms that others can use it, right? So we've got to have people start talking to each other. We can't have all of these different islands where we can't take that because a patient initiated it. Well, you know what? It was a health exchange that grabbed that data right from the institution. We didn't touch it, right? Well, but maybe it's not an assembly of everything we should know. Well, who's gathering the data from Wichita, Kansas? Because those kids are told to go home and make memories. They're not in any data system. If you're not in a partner with CBTN, if you're not in a PNOC hospital, if you're not in a PBTC hospital, your data is probably not getting counted in DIPG because there's not very many COG trials out there. So that is my issue. Like, we can't think that one lane fits all, but we do have to work together to build the best lanes, right? And I have yet to find, and there's a lot of people that know more than me, I have yet to find somebody that can say, here's where it is. Here's everything there is. But I have had conversations with a couple that say, we've been given a $2 million grant, so we're going to have students take data points out of records by hand and put them into an Excel spreadsheet so it can be shared into a data system. Does anybody see any flaws with that? We're going to give institutions $200,000 an institution to do that. Now, this is where I have a question, not an answer. I'm an attorney. We can do artificial intelligence, natural language, and transcribe a 40,000-page trial in one day. We can outline it, annotate it, and you know what? We do not do that with our paralegals anymore. It's not the best model. We do it with technology, and we have our paralegals go back and fix what may not be right. You guys, we have to come together. We have to come together. They're wrapping me up. Thank you so much.
We will have a 10-minute break, so if you want to kind of chat with them, there's the snacks outside in the foyer.
Hi, everyone. Welcome to the Advances in Omics session. This was the best, like the most fun session to put together for me. I am really a genomics geek. So I'd like to introduce lead panelist and moderator, Dr. Adam Resnick from CHOP. We have Amanda, sorry if I butcher your last name, Polowich? From Seattle's Children Fred Hutch Institute. We have Dr. Carl Koshman, a veteran in DIPT plays from University of Michigan, and a personal friend and a great collaborator, Jay West from BioScribe. Thank you.
So Misha charged me with sort of setting the stage for what's going to be an awesome panel discussion, and we hope we have engagement and questions. So much of what we talk about in the context of a disease setting where there isn't a standard of care is really generating more data to address that lack of information. So Misha said, can you provide the context for what is omics? So let me start with, just to level set, what do we actually talk about when we talk about multi-omics? And so, like everyone, I go to Wikipedia for the source of truth. And reasonably, they define omics as a collective characterization and quantification of pools of biological molecules that translates into structure, function, dynamics of an organism or organisms. What does that really mean? What it really means is what you've heard about in multiple contexts, is the kind of data we can generate typically from samples. And so the methodology that most of us pursue in this context is the collection of highly curated samples at the time of surgery or a biopsy, generation of data from those samples, and I'll talk a little bit what does that omic data mean. But just generating data itself is not sufficient. You have to transform that into information that's queryable and ultimately into knowledge, either knowledge about that patient or knowledge about a disease in a way that can then drive impact. And you'll see some logos at the bottom that says like platforms and communities that are connected. And this partly connects to, if you were here to the previous session, the need for systems that connect and intersect across these resources. But all of this really boils down to a key premise for neuro-oncology, but for cancer and disease as a whole, is that in order to make progress, we have to create measurements. And those measurements are the only things that can drive understanding. And it's understanding that opens the door for action. You can sometimes get lucky without understanding and go from measurement to correlation to action. But you can never drive action from a space of something not working and not understanding why, right? So you can get lucky and sometimes something's working and you don't care why, just keep doing it. But by and large, in the disease landscape where you don't have that luck, right, the only way to get to action is through measurements and understanding. And so why and what do we measure? You've heard through most of the previous day and today that there are measurements that take place in the context of clinical care, right? You get assessment by clinicians, you get radiological measurements, you get a pathology measurement. Here we're talking about these omic measurements and what they mean, but in reality, you then have to integrate all of it to drive a view of a patient. And that view essentially allows a clinician or a scientist to say, I can understand how this patient relates to other patients, or maybe does not relate to the other patients. I can potentially predict how they might respond to a drug or not respond to a drug, and potentially define a course of care. The challenge for us is that cancer is complex, not just any one cancer, but all cancer. And this is essentially a set of hallmarks that we as a cancer biology community understand are ways that cancer subverts normal processes in order to thrive, right? And you can see that this is a wheel with many colors, right? So cancer has a very large palette of solutions in order to progress, advance, and do what it does. And all of these solutions are driven by molecular mechanisms of change inside that tumor. And so by and large, omic-based approaches are trying to characterize the mechanisms by which cancer is harnessing these solutions, because there are multiple ways to get to the same solution. And those omics approaches then provide you a snapshot, typically, of how to understand the current state of the tumor in ways that might be predictive. And yesterday and today, you've heard that those omic approaches really take place at these three different biological layers. Right? They take place at a genomic layer, at your DNA. Every single one of your cells in your body has the exact same DNA, but that DNA is activated and modified in different ways to support your normal biology, but cancer harnesses those 3 billion letters in your genome in multiple ways. Across that genome, you have 25,000 genes that, again, get activated differentially, and cancer, again, differentially modifies those different combinations of genes and their activation at the RNA level. But ultimately, proteins are the functional components of cells, and that's where most drugs act, right? So most drugs bind to and act at the protein level. So many of you are familiar with, you might have gotten a diagnosis. For example, if you DIPG, you might have, someone might have told you have a K27M mutation. What that really means is that there's a protein expressed in a cell, right, that has a lysine that's been changed to a methionine. And that protein then has a different function than it was designed to in the cell. In order to understand that, we have to take measurements across all these layers, assemble them, and then create a view. And so this is the modern era of multi-omic analysis, is to create these complex layers that essentially measure different views of biology and how they get assembled into the mechanisms by which cancer is operating. And typically, two things happen when we do that. One, we can ask the question, can we explain the biology of a tumor class? We can potentially say, we think that sonic hedgehog medulloblastoma, for example, that first part that said sonic hedgehog is really a classification based on an omic analysis. So we can say, okay, you belong to this class of tumors. But then we can also potentially try and predict which drugs might be appropriate for you, which drugs might bind to certain proteins or alterations. Or hopefully, if there aren't drugs that exist, we can say there should be a drug that targets this protein or this alteration in that context and define a clinical trial. Brain tumors are special. They're special in a complex way. And we'll hear a little bit as we talk about this. In contrast to almost any other organ system, the number of different cell types in the brain is order or magnitude larger. And so when we say a brain tumor, most of you have heard we talk about more than 120 different types of tumors. The reason there are so many different tumor types is because there are different cell types. So you might have heard, for example, you might have a glioma. Right? That first part of glee really represents the fact that it came from a glial origin. Or if you had a germinoma, it's because the first part is germ, it means it came from a germ cell tumor. So only certain tumor types can come from certain cell types. If you have more cell types, you'll have more tumor types. It'll be more complex and more challenging. That complexity, right, of multiple cell types, right, so you can even do some rough approximation of a challenge. 5,000 different types of cells, 25,000 different genes, 3 billion different letters in the genome, and the diversity of humankind, right? That layers of complexity is what drives the challenge for defining mechanisms and cures for brain tumors at large. The way we try to do that is to create atlases that support an initial understanding of the search space for what it means to be of a certain tumor. And these atlases are defined by creating large-scale data sets from collected samples across multi-omic data sets. But to operationalize that atlas on behalf of patients is the next challenge, right? And that's partly what hopefully we can get the team on the table to discuss. And I'll start with Carl. So Carl and I know each other very well. Carl, I'll start with you in part because one question that comes up to us very often is, what assay should be done for a patient when they are going to get diagnosed, right? Because the amount of tissue might be limited, and there might be decisions that a patient has to make. And in general, that immediately brings into context questions about, across all this omic space, can I actually do any omic analysis I can? Or are there some assays that have to be performed certain ways or not in order for clinical decisions to be made? So start there.
No, that's a really good question. And I guess that's sort of a core discussion that we are frequently having with our pathologists, clinicians, and other researchers. And I think specifically as it pertains to DMG and DIPG, almost always we're talking about a biopsy and not a resection. So we're having to come up with a plan for a very small amount of tissue. What we talk about as the key thing is we need a quick diagnosis, because that's going to be able to make plans in real time. And so the pathologists are key to that small amount of tissue being used clinically for stains. But actually, we've made a lot of progress with pathologists, at least at the bigger brain tumor centers, of not using a lot of tissue at that point, not going into assays that are probably more based on brain tumors generally, especially like adult brain tumors, where you're going to quickly run through the small number of biopsy cores that are there. What we really want to do is just couple stains, a couple slides to figure out what the tumor is. For midline glioma, is there a histone mutation, and then stop. And then it is really getting into sequencing. And at this point, we would say that it should be standard of care, that there's DNA and RNA sequencing. And there's a whole different discussion we could have about whether that should be panel or whole genome. I think we've made a lot of progress with MyOncoSeq and CBTN and PNOC of using very small amounts of sample and still getting both panel and whole genome. And the panel is really useful in real time. Those are the actionable mutations that we can find and act on and might push you towards one trial or another. The genome, at least in its current state, is useful for discovery. And the more I think we're quite honestly just stepping onto the landscape of what we can do with whole genome. And so that's really, if we talk to families, that whole genome is probably more for helping future patients than helping that patient in real time. But if you can do it without using any more tissue, then that should be, I think, a part of the process. Then you get into a whole nother layer that I guess you started to look at in terms of, are there things that we could do with methylation or single cell or proteome or metabolome that might change management? And I think from a research perspective, we want all of that. But for that patient, is any of that changing management in real time? And at least right now, with all those tests being in sort of a non-CLIA certified setting, we're really not using those clinically. But I think as those tests get better and as we publish more, that maybe there is an assay within single cell. Maybe we can use the same amount of tissue and get this single cell level information and have that start to adjust management. I think that needs to be published before we go there. So where we're at right now, just to summarize, I think would be, we want to maximize sequencing with a small amount of tissue, leaving that patient with more tissue for potential future trial. That's it.
Yeah, let me pull a thread of principles from what Carl just said. So the first principle is when you don't know something, right, you're trying to maximize the amount of data that you can generate, right? So in the absence of knowledge or understanding, your job, typically as a scientist or as a clinician, right, is to generate as much possible information, because you don't know which information is going to be the one that you need to use, right? The second principle, which Carl mentioned, is the minute you do know that one piece of information is important, that piece of information becomes prioritized. And that piece of information is prioritized in the context of omics, oftentimes gets asked as rapidly as possible and with a selected view, right? So oftentimes when we say omics, right, we mean, let's say, the entire whole genome, right? There are 3 billion base pairs. But in Carl's example, as a patient comes in and is getting diagnosed, let's say for DIPG or DMG, he might only need to know about 5 or 6 genes, right, to move to the next decision space, right, in that context. And so that's the tension, right, that a community like ours faces, is that as we gain new knowledge, we have to prioritize making sure we capture that. But we also know that that knowledge is not sufficient to drive new cures or therapies as of yet. It's just a decision point at this moment in time for patient care. Carl mentioned the word CLIA. For that, I'll turn to Mandy to address that. So Mandy and I actually connected through an NCI-led initiative that focused on proteomics. This was an NCI initiative that tries to create large-scale atlases of the protein constituents, initially of only adult cancers. But Mandy, when I first approached her and said, would you like to work on peds? She said, hell yeah, I want to work in the pediatric space if there's an opportunity. And you didn't hear Carl even mention proteomics in the context of a decision-making space, right? If you had a proteomic assay, how would you use it for a patient? The exciting part is that I think folks like Mandy are beginning to ask the question on how to do that. But the first requirement, which Carl actually highlighted, is this term CLIA. How can things become CLIA, and what does that mean?
Yeah, thanks, Adam. Sorry, I have a mask on because I'm going to go visit my immunocompromised father, but if you guys can't hear me, I'll take it off for the purpose. Just let me know. So yeah, I'm a clinically trained oncologist. I actually have a PhD in genetics. I spent years studying genes and gene expression and how they affect how cells behave. And I became interested in proteomics 20 years ago because of the experiences I had with my patients. One patient's tumor would melt away with standard of care treatment. The next patient looked like they had the same tumor and their tumor wouldn't respond. One patient would get toxicity from chemotherapy. The next patient I'd give the same chemotherapy to would have so few side effects, they'd express concern that they weren't getting enough of the drug. And so this diversity was just clinically apparent on a daily basis, and it became ethically difficult for me. Yes, I'm following standard of care, but what we needed was better diagnostic testing. And having spent a lot of years studying genes and even doing postdoctoral training at one of the major genome centers as the human genome sequencing was being completed, it was clear that the genome wasn't going to provide all the answers. It's going to get us partway there, but not all the way there. And that's borne out to be true. We all want precision medicine. We had a session yesterday about subtyping tumors into many, many different subclasses to try and figure out how to better treat them and match patients to therapies. Well, to do that, not only do we need the drugs to target those, but we need diagnostic tests to figure out which patients go into which categories. Currently, precision oncology is genome driven. We sequence patients' tumors and we try to predict what drugs the tumor will respond to. That's gotten us a fair way down the road, and for a handful of tumors, it's actually cured patients. But for the vast majority of cancer patients, the response rate to the genomically predicted therapy is between 20% to 50% if you're lucky, and the vast majority of those tumors recur later as resistant disease. So we've been doing this for a lot of years now, and that seems to be how far we've gotten, which is a fantastic advance. But I would say one of the second greatest contributions of the Human Genome Project was providing us with the genome sequence, which is the blueprint for making all the proteins. And that has enabled this new field called proteomics. And as Adam mentioned, the genes are the blueprint of everything the cells can do, but the proteins are what actually do the things that the cells do, and the proteins are what the drugs are targeting. And there's many layers of regulation between the genes and the proteins. So if you're only looking at genes and trying to predict what drugs should target the proteins downstream, there's a lot of missing information in the middle. And that's why I became interested in proteomics 20 years ago, and I've run a translational proteomic lab at the Hutch ever since. And Adam mentioned CLIA. Well, there's two different types of proteomics. There's an untargeted mode of proteomics where we use a mass spectrometer, which is a machine that we can use to look at proteins. And we try to see everything in that patient's biopsy. And we do that across hundreds of patients. And we look for patterns of protein expression, just like we do with genes or RNA, and try to find patterns of proteins that correlate with disease phenotypes, like outcome or survival. And then there's a targeted form of proteomics where we've already done that discovery and we've narrowed it down to a handful of proteins we think are important, and we want to measure those in a clinical setting to help guide clinical decision making. That's a completely different level of rigor that generally occurs in a regulatory setting, such as CLIA. And CLIA is really a collection of laboratory requirements that sets a standard, a high standard for performance for running tests. My lab is a CLIA environment for running targeted proteomic tests, which are trying to move now into the diagnostic space. We've had one success so far all the way through, and this is a targeted protein assay based on mass spectrometry for the protein thyroglobulin. This is a cancer biomarker for thyroid cancer patients, and the traditional assay used in clinical labs is inaccurate in about 15 or 20% of patients because they make autoantibodies in their blood that interferes with that measurement. And this assay that we built can circumvent that. And that assay is now run in clinical reference labs in the US and Canada in patients who make these autoantibodies. So it is true that proteins are behind DNA, and DNA had a big head start on proteins. The proteomic field got started quite a few years later, and also has the disadvantage that we can't amplify proteins with PCR like we can DNA. You can take a tiny, tiny amount of DNA and amplify it up. And so that's definitely an advantage over it. But the protein space is critically important because that's what the drugs are targeting, and that's what's carrying out the functions in the cell.
Again, I think you heard from Mandy that refrain, right? That once you create that atlas and you understand that these are key proteins that you need to pay attention to, then a natural order of events is to develop a clinical assay that can then be run on behalf of patient care. The challenge for a community like ours is that we still haven't defined what are the things to measure. So we're constantly in this battle to generate that landscape view as we're trying to help inform patient care. And so, as Carl and Mandy mentioned, right, a critical decision space is really how do you maximize the use of those samples? And the part that's super exciting is, and this is true, like for example, when Mandy and I first started, you had to have like a really big chunk of tissue, right, to generate even just that landscape view. And over the past 3 years, suddenly that tissue amount got smaller and smaller and smaller. The same is true for DNA sequencing or RNA sequencing. So the technology space is allowing us to do more and more. And here's where I'll turn to Jason, because it's gotten to the point where we can actually start measuring individual cells. That's something to keep in mind, is that typically, right, you take a piece of tumor out. That tumor is actually millions or more of cells. Each of those cells is sort of, yes, it pays attention to its neighbor, but it's doing its own thing, right? That cell really cares about itself as a cancer cell proliferating and multiplying. It's paying attention to its neighbor cells, but there might be differences between those cells. So you might have heard a term tumor heterogeneity, right? Or if actually when you look at that MRI and you see that it's not just this uniform shape and color, right? It looks variegated. That MRI is a signal of biology being variegated in that tumor. And so single cell technologies have emerged as another layer of omic data, right? So to date, the majority of assays have done what is called bulk sequencing, where we essentially take all those cells, crunch them up, and treat it as one sample, and average all the signals of those millions of cells to derive features. But there's recognition that all those cells are not equal or of the same cell type. With that as a premise, Jason, perhaps talk a little bit about BioScribe, the vision behind it, and how single cell technology you feel, might change the landscape.
Yeah, thanks. So, Mandy, your comments are great. I think we are a company that focuses on the amplification of individual cell genomes, and the reason we do that is to understand the foundation of the disease. But the omes in isolation are maybe informative, but not incredibly helpful. And so, and I'll give a specific example. So, actually, before I do that, while we optimized and perfected our ability to, analyze individual cells and their genetic foundation, we realized that, again, in isolation, the information was somewhat limited. And so, like we were saying, it's proteomics, it's transcriptomics, it's methylation. And so we began to integrate all those measurements into the individual cell. And that gives us an amazing view of not only the foundational, the genomic foundation of that cell, but also the functional consequence of the changes in that genome. And that, as Adam mentioned, is that each one of the cells in those tumors are different. And they're not all tumor cells. They're mesenchymal cells, they're immune cells, they're, 5,000 different cells in the case of the brain. And so even being able to pin down that cell type through the RNA signature to say this is, a glial cell, for example, and it has a deletion or an amplification in this portion of the genome, and those genes are either upregulated or downregulated. And so just to give a bit of an insight into this, which you guys might find interesting, is, we were just finishing up a cohort of patients, adult cancer, and we found evidence of ploidy. Ploidy meaning there's a portion of the genome that is amplified or a portion of the genome that's deleted. And we correlated that to the gene expression. And so if I have an amplified portion of the genome, meaning that it's got an extra copy, you would expect that the expression of those genes goes up. And in many cases, it does. And conversely, if there's a deletion of a gene, that the gene expression goes down. And we found cases like that too, but we also found the converse, where there was increase in ploidy, meaning there's an amplification of the genome, and there was a decrease in gene expression. And decreases in ploidy and massive increases in expression. The point is that it's pretty complex. And if we're gonna approach it as root cause analysis, it's just understanding the disease, as you mentioned, Adam, is we took an approach to just look at these individual cells with unbiased data. And we generate an amazing amount of data that is discovery driven. It's not yet diagnostic, well, not entirely. And this is one of the questions I was going to ask back to you, is, we, as I mentioned last night in my kind of remarks, we've just finished this cohort of patients, and we were able to, I guess unfortunately, redefined, the trajectory of these tumors. And in some cases, and being somewhat controversial here, indicated that the therapy was not optimized. And we're not really sure what to do about that clinically. And we need help to understand how to translate this into the clinic, because it's so much data, and we know for a fact that we can affect the outcome of the patient, but we're not sure how to, translate this back to the clinic. I don't know if that's helpful. I hope that's helpful.
Yeah. Let me pull on a couple threads, but then would love also the panelists to answer. So you sort of highlighted, the immense diversity that you essentially encounter when you start measuring things at a more and more refined level, right? If you're starting to measure each individual cell in a tumor, you're going to see that each one of them is different, but somehow the system together acts as a whole in the context of the tumor, right? And the interplay between them. I want to just poll and make sure the audience, has the same sort of shared understanding. So when you essentially say, you might have a ploidy difference, or, a whole, Elaine mentioned yesterday, a whole chromosome might have been duplicated, a whole part of your genome, right? So you're born with 2 copies of each one of your chromosome. Cancer, essentially, one of its mechanisms is to just make copies of the parts that it potentially cares about, to regulate the availability of essentially material to make genes. But then you highlighted that essentially, even though there are multiple copies, cancer was smart enough to say, actually, even though there's an extra copy of this entire piece, I don't want extra of this part or this part, so I'll actually downregulate those pieces. And I think that's a key tenet of cancer, right? That each layer, cancer will modulate. And if you don't measure how cancer is modulating, you make assumptions that potentially are not reflective of the biology, which I think is what you're trying to highlight, right? That I could assume something by just measuring the DNA, but until I measure the RNA, that assumption is potentially false. And that ties to what Mandy said, which is I can measure the RNA, but until I measure the protein, I can give drug all day long based on my RNA result, but if the protein's not there, it may also be false, right?
So my partner is a pediatric oncologist and had been doing bulk sequencing for a long period of time and didn't see a positive outcome for his patients. And so we began sequencing individual cells because of that. Because essentially, what we see is just, yes, increased resolution, which creates a lot more data. But in terms of mutation load, for example, we see a massive increase in mutation detection just because we're starting to separate all the noise apart. And so our resolution, I guess the analogy would use is when we're looking at a tumor and its genetic changes, we're just increasing the objective strength, where our microscope objective is just a lot stronger. We go from 4x to 100x. And so I hope that answers your question.
Yeah. And then let's take your question back to both Mandy and Carl. So your basic question was, I'm applying a new technology and I'm seeing these results. How does that actually come into clinical care to change the decision-making process, right? How does that go from that research setting into a CLIA setting or decision-making space? Mandy, I wonder if you can just comment on what's the arc of that for proteomics by analogy, and what are the challenges and opportunities that you faced in that setting, and what does it take to actually go from a research measurement setting to achieve adoption of a new technology for clinical care?
It's a fantastic question, and probably the most important thing we're going to talk about in this room today, because we really need advocacy. So in fact, I'm at a crossroads in my career because of this. So let me give a brief example of a study we just published that illustrates the problem. So this study focused on ovarian cancer, which in general is a very chemotherapy-sensitive tumor. But 20% of patients have a tumor that does not respond to standard chemotherapy. Their tumor grows while the patients get chemotherapy. And this is essentially a death sentence because those patients get so sick. They already had advanced stage disease. There's no good early detection of ovarian cancer. They get so sick getting chemotherapy that's not working for them, because we didn't know ahead of time it wasn't going to work for them, that they no longer are healthy enough to participate in clinical trials. No research can be done to study this problem, and for 40 years the outcome hasn't changed for these patients. So using these new omic approaches, these atlases like everybody's been talking about, we discovered a 64 protein signature that will predict, before any treatment, about 35 or 40% of the patients whose tumors won't respond. Now, this is a problem that people have studied for a long time. It's a hard problem. There's 30 years of scientific papers published. 900 genes have been implicated in this process, and not a single one of them has been translated to a new diagnostic. Why? It's technology limitation in the case of proteins. Usually we use technologies to measure proteins clinically that measure one protein at a time. And if you have 900 genes that encode at least 900 proteins that can affect whether your tumor is going to respond to this chemotherapy, and your technology lets you measure one at a time, you can see there's a massive mismatch, and nothing gets translated into clinical use. And all of these things get tested one at a time over the last 30 years, and everything is said to not work because it only picked up a couple % of patients. And what we need is something that can measure a lot of proteins at once. And so for the past 20 years, that's why I dove into this proteomic field. And we have developed and clinically implemented now a platform that can do that. The problem is no longer scientific. Okay, this is where advocacy is needed. We all want precision medicine. We all want to know what subtype of every tumor we have and what's the best treatment to match. And our NIH funding and almost all of the commercial funding goes to developing new drugs for those subtypes of tumors. And that's great and that's important. You hear everybody talking about the next targeted therapy. How many people do you hear talking about the next generation of diagnostics that are truly going to be needed to realize the potential of precision medicine? Almost nobody. You know why? I know this because I've been struggling with this in my own career. What am I going to do now? We already solved this problem. I can sit here and churn out these atlases until I'm, headed to my grave. But how's it ever going to help patients if we can't translate that new information with new technologies into clinical use? In my opinion, what I've seen so far, it's an economic problem. It's capitalism, which is great, but capitalism rewards profit, and diagnostics are grossly undervalued, they're grossly under-reimbursed, and if you're a VC firm, and I know this because I've talked to them, Would you rather invest in a company that has a 1 out of 10,000 chance of making the next blockbuster drug like Lipitor, or a diagnostic company that might have a great multi-omic, the next generation are going to be multi-omic AI-driven tests that take into account patient characteristics, all of the blood tests, the imaging, and these new molecular profiles. And it's going to be algorithmically driven. And there's not a large profit motive to do that right now. Diagnostics are high volume, low yield in terms of profit, and it's very difficult commercially to move things like this forward. It's no longer a scientific problem.
I just want to add, on the very similar vein, to discuss some work that's actually supported by PBTF as well as DARPA. We had developed a digital droplet assay to look for the histone mutation in blood and spinal fluid. And after like 5 years of work, we could find that needle in the haystack each time reliably. And so we published it, and then we published that it actually helped show us whether patients were responding to the ONC201 treatments. And in my, sort of not having a great understanding of how something like that works, I figured, industry would take the test from there. We purposefully did not patent what we came up with. We wanted someone to take it from there and start offering it clinically. And it just like crickets chirping ever since we published. So we went to University of Michigan, which has an M-Labs, and we went to they have a business team, and they basically need to pay for the costs of developing and maintaining the assay. And when we told them, even when we kind of upped our numbers of how many samples we would get, they said, you're not even close to covering the costs of developing and running this assay in a CLIA setting. So at that point, we went to foundations, and that has actually gotten us really close to the finish line. So we think with the funding that we now have, really what it pays for, just like where does the money actually go, is procuring a lab bench in a CLIA setting on a very busy and congested campus where there isn't that space. But we now have the space. We have to have machines dedicated to the test, and we have to have technicians working on it. It's not going to be a profitable test, but I think a lot of the families that are in this room who read papers like that and say that sounds great. I would love to have my child's samples get these tests in real time, are frustrated to see that in fact we're still far away from running the test. But I think the hope is that in 2024 this will be a CLIA certified test that we can run on patient samples, and it really took family foundations to get us to that point.
And then the question is, who's going to pay for that? Is it get reimbursed? Because insurance companies or CMS don't just pick those things up and pay for them. That's a giant battle downstream. Diagnostics accounts for a fraction of healthcare costs, and yet over 80% of healthcare decisions are based on them. And there's no major, and it's not going to happen in the commercial sector anytime soon. And the government should be doing this, and we need advocacy. This is a great project for ARPA-H, right? They want things that are too risky commercially, that are transformational, and that cross government agencies. This fits that bill perfectly. I contacted them, I was told, write a white paper on that so it can be socialized. If anybody has time to write a white paper or experience writing a white paper, it would be lovely to team up with you to actually do that, because it's not what I do for a living and I don't have time, but I would love to participate in that. Yeah, it's a terrible problem. It's not scientific anymore.
So I promise you, we did not actually coordinate the structure of this talk, right? But it's exactly where I'd hope we would end. Because, we started with this question of, you have precious samples, what assays do you do? And I was hoping we'd get to exactly this context where essentially the promise of multi-omic analysis is right in front of us, right? We have examples of how that multi-omic data can be harnessed and actually applied. The challenge is that to transition and leverage that research setting and then apply it at the clinical interface has an extremely high bar of financial and implementation challenges. We are undergoing the exact same process at CHOP, right? We were trying to launch a test clinically in the pathology lab. They told us it would take 3 years and they'll have to do an assessment of the budget. The only way, right, to actually do that is to create a separate laboratory, right, under the CLIA regulated environment, and then link it to patient-driven resources and foundation that says, okay, we're willing, right, to support that framework, because otherwise the conduits are just not matched. So I think that's the real opportunity, to intersect between this space of emerging knowledge and its application and how patient-driven, informed patient-driven context can intersect. Now that's not easy, right? Even for me, as I was preparing this talk today, right, I'm trying to think and level set, to Lisa, who says the last time I took biology was in 1988, and no matter how smart you are, right, if I start listing the 100 different assays that one can run, right, it'll take some time to figure that out. And so this is where we have to have a really tight partnership, right, between the research setting, right, and the advocacy setting, a trusted partnership, right, that essentially says, yes, we as a community agree that this needs to move forward. This has potential, right? The liquid biopsy for histone mutation or proteomic assays that predict efficacy or a single cell assay that can actually, define diversity and link it to outcome. I'll turn back to Jason because you actually made the right turn in the path when you essentially laid a question of, all right, I can do all of this, how does that get implemented? How are you guys as a commercial entity now, right? Neither Mandy or Carl on the commercial side, how are you guys thinking about taking the next step?
So, we obviously operate in a couple different markets. And so let me tell you about a massive dichotomy. One of the markets that we've done very well in over the last 3 years is in pre-implantation genetic testing. It's a cash-based business, and they value the test, and they pay for it in cash. And so these IVF cycles are sometimes up to $50,000, and they'll be paying, our clients, up to $10,000 for a genomic test on just a few cells that are extracted from the trophoblast. And so, if you compare that to, what's happening, there are so many barriers, but I would say something entirely confidential. In a cash-based business where somebody really cares about the endpoint of their pregnancy, they're willing to pay thousands of dollars for these tests. But because of the infrastructure that's laid into an oncology test, it's quite different.
Yeah. And that's a risk space, just to call this out, right? We talked about Lisa. Every time I hear her talk, it says, how can I make sure Wichita, right, is equivalent to a Philadelphia in terms of the opportunity landscape, whether that's data or anything else, right? The minute you essentially isolate opportunity, right, to a cash-based setting that's dependent on individuals, you'll create dichotomies, right, of potential access, right? And that's the last thing you want in a rare disease population where you're actually trying to create a coherent framework of an approach, right? So that's the risk space and the differentiator, right, that we have to engage as a community in that setting. We have 5 minutes left, which I told myself we'll leave 10 minutes for questions, but I only got to 5. So let's pause, and then anybody ask questions or comments?
Raise a hand, and I'll come with a mic so that our live streamers can hear.
Hi, I'm a bereaved mom, DIPG. My name's Bonnie, and advocacy is huge. I hear you. My question is, and I'm not even sure how to get it out, but I think, parents like us who would have loved some kind of diagnostic tool for our children. I mean, 10 years ago, Tatum had nothing, we didn't do biopsy, anything. So right. How to advocate for that? What do we ask for? That's the thing. Because there are opportunities where we do have people who are listening. And so I think, and I'll be honest, I'm one of those people who's like, yes, please give us research dollars, research drugs, we need them. On a side note, I have a special love for pathology, so I don't know how to ask for that or where that falls. Yeah. And I don't know if my question even makes sense, but
I think you're asking,
guidance is my...
you're asking essentially like what's the action we're asking, right? Mandy essentially highlighted what you heard. I think I'll distill it, but then please, you guys chime in. One is in spaces where the economics don't make sense is where the federal government has to step in. Right? Because you cannot count on market economy to drive that process forward. the government stepping in can drive an economic framework, right? So it doesn't mean that the government comes in and only supports academia, right? The government can create policies, they can create funding sources. Both of those can drive economic change. For example, you can imagine a policy space that says this has to be done and be covered by insurance, right? And then that essentially creates an economic framework for that. Or you can actually say this has to be paid for, right, at least for a period of time, so we can define, its impact, right? Those are two contexts that the policy or financial space that the federal government setting has. But the other part, which I think you heard, is we can't always count on the government to move at the speed, right, that we expect. And in general, our community can self-organize and pool resources to advance and demonstrate impact. I think that's the other opportunity, right? That there's shared understanding in our community about what's required, right, from a diagnostic or an omic perspective to go to the next phase. Maybe there's an opportunity not just for one foundation or two foundations, or maybe several foundations to say, actually, let's pool our resources and invest in this. This is where the coordination, right, of support, all right? And this is something that potentially is not disease specific, right? It's a shared unmet need across multiple brain tumors. Those are the kind of opportunities that can be extremely rich, right? Where essentially, if you start solving the problem for DIPG, you're solving it for medulloblastoma, you're solving it for others because it's a common framework problem. I think those are three ways, essentially, but open to additional guidance from you. Mandy or Carl?
I don't think the government understands. I don't even just think it's that they're slow. I don't think they understand. After 20 years of doing this, I'm an oncologist. I do this to get us back to patients. We've solved the science, we know how to do it, now we just need to implement it, and we're up against this wall. So I've talked to a lot of people. I've talked to advocacy groups, I've talked to outreach groups, for example, for the American Association of Clinical Chemistry, which is where the clinical pathologists are. They do the lab. Those guys have advocacy groups, but you know what they're advocating for? This argument between should all lab tests be FDA regulated versus should laboratory developed tests be. That's what the government thinks diagnostics is right now, because that's what all the advocates are talking about. This is a different problem. We want precision medicine, we want precision oncology. That doesn't just mean drugs that are going to bankrupt all the patients. It means good investment in good diagnostics, and not the way we've been doing it for the last 50, 60 years. We need real next generation diagnostics that actually captures the complexity of these clinical phenotypes that we're seeing in patients, which means using these new technologies and figuring out how to incentivize that commercially. And then once it's incentivized commercially, it will take off, right? I mean, this is what capitalism does at its best. It is not incentivized. It's a big problem.
Right. So when we spoke about in the beginning of the panel around this notion of like, if you have limited tissue, you have to decide what assays to run. But if the decision space is not based on how much tissue you have, but on the financial support for it, that's where there is a moral mismatch, which I think is why.
And there's fear for pharmaceutical companies. We work a lot with pharmaceutical companies, and they know this. They know that the diagnostics they're using aren't the best. But do they want their blockbuster drug that they've spent hundreds of millions of dollars developing and it's going into its pivotal last phase clinical trial, do they want to bring that in with a novel, multi-omic, next-generational diagnostic test, or do they want a little IHC test that measures one protein like people have been doing for 50 or 60 years so they know how to get that through the FDA?
What Mandy is pointing to, and it's a conversation we've had multiple times, is that essentially drug companies will do the safest and least controversial action, right, to make sure they get approval, right? They have to, right?
Yeah.
And that's because they're orienting towards the regulatory framework. If the regulatory framework says you have to have a companion diagnostic that only measures this one protein, then that will never come through Mandy's lab.
Okay, I have just one question. This is an excellent panel. Thank you all so much for that. My question is just, if it's something we have to ask for other than dollars, right? We know that dollars are something we want, but if we can't ask for dollars, and what we really have are some buy-in from, some tech companies, some data companies, et cetera, that might say, let's make us your use case. Let's see if it works here, because then we can shrink wrap it for others, right? What are we asking for? What are the top 3 things you need to do your work? Is it a change in policy between academics and industry? Is it, I mean, what is it you need to use things that are maybe out there and fit them for pediatric brain cancer best? Thank you.
I'll go first. You know, I think, you know, we focus a lot of our research dollars on patient samples. And so I was just talking with somebody this morning about this, is that, you know, you know, each patient is very different. And we know this because we sequenced, you know, 15 different patients and they're for 20, and they're all different. And what, you know, if we go down the standard clinical trial path, we're going to say, okay, you know, we're going to group all these people together as if they're one, and they're absolutely not. Like, that drug will not work on that patient. We know this, and so I think what I would ask for is a mechanism where we can use that data effectively to actually personalize the medicine, because otherwise, as Praveen was talking about this morning, we're not going to be able to use these combination therapies. I don't know how we get to this. So that structure has to be different. I just don't, that's something that needs to be solved for us, because again, we started it to have impact. It wasn't, you know, yes, we have to do all the commercial things to keep paying our employees, to keep doing all this work. That's a necessary component of running a business. But in the end of the day, the whole idea was to have a dramatic impact on oncology in writ large.
I think, Lisa, your thread is an important one, and that is that, you know, these are innovative, leading-edge technologies, right? They will have application in broader disease space eventually. But I think the narrative that you pulled on, which is that the pediatric cancer setting, pediatric brain tumors, can actually be a hub of innovation because we have a coordinated framework of shared prioritization, alignment, advocacy, mission, funding sources, right? That is an extremely unique setting that does not exist broadly across cancer as a whole, right? And I think is what Mandy is essentially challenging us to say is, you as a community have a unique capacity to voice, right, these challenges at the policy space, financial space, and implementation space, right, in diseases that have no standard of care, no pharma-based investment in, right? We could lead diagnostics, right, in ways that then brings those in. At least for me, that's the exciting opportunity there. Right? That's the other side of the coin where we are. It's an opportunity for us to be the innovative hub for implementation, because the parents will do more for kids than adults will ever do for themselves, right? So you will take any assay, right, to make a decision, and you have to create that challenge, you know, as a space to move forward. All right, they're telling me to wrap up. Thank you guys for the question. Thank you especially to the panelists for the discussion.
All right, well, thank you everyone for attending. This has been a really exciting conference and opportunity to meet lots of great folks. So today I'm going to tell you about what we've built with XCures. So we're a health data technology company, and we've been around for about 5 years. A little bit of background on who we are, where we came from. We were spun out from a nonprofit patient advocacy organization that essentially helped to navigate individual patients with cancer through all the massive amount of scientific, medical, clinical knowledge to help make the best informed treatment decisions. And so, what we've done is really built this platform to enable access to medical records so that those scientists could help to navigate those patients. But also we've done this in the context of clinical research so that all the data that we collect can be repurposed, shared with researchers, shared with clinicians. And so you'll see the puzzle pieces. We actually have a puzzle that we're trying to put together at our booth. Unfortunately, there's 500 pieces, and each person had 2 pieces in their book bag. So if everyone can bring them together, we might actually get like a third of it done. So, and so, what we have is really a direct-to-patient precision oncology clinical research platform that we've built. And it's all under the umbrella of Excelsior, which is it's called an observational research protocol. And it's a master observational research protocol. So we're able to sort of aggregate data across multiple different cancer types. It's totally virtual. Patients can simply go online, they sign the e-consent, and they sign a HIPAA release. And then we do everything else downstream of that. We don't need patients to continually fill out surveys. We don't need them to, continue to work with us to, tell us if they've moved centers. We can follow patients prospectively through that. And so this is really a unique platform that we've built because we're sort of a third party. We're not affiliated with individual institutions, so we can sort of stand separately and then aggregate all the data from all different institutions. And this is a different approach than you've heard Adam and others talk about, where they work with individual institutions and they get everything out of that institution, which is extremely useful, extremely important. But we've taken the opposite approach, which is work with individual patients and get all of the information from all the places those individual patients have been to, and then we stack that together, create cohorts of hundreds of thousands of patients. And so what we do downstream of this is we do essentially clinical data annotation from those medical records that we've gathered. And so this is essentially called EMR to EDC. So we're taking all these electronic medical records, we're creating clinical trial grade data from just the standard practice of medicine. And the last thing that we've done is essentially layer on other protocols, other research opportunities for patients, bringing studies directly to patients so they don't have to be at a large academic medical center. All of this can operate within this platform. And so it's a really unique approach that we've taken, very different from institutionally driven clinical research. And so this slide's a little complex, but it kind of goes through the data processing pipeline and what we do. I'm going to walk through it pretty quickly. Essentially, we pull all of this data in together. We get it from EMRs, from imaging centers, from the next generation sequencing labs, faxes and emails when we can't get it electronically. We bring all of this together, annotate that clinical trial database, and we create a complete longitudinal cancer history for every patient that comes onto the platform. This is a little bit about our technology. Just really quickly, we have a way to highlight through everything. All the elements that we annotate, we can trace right back to the source. This is really important for clinical research and for ensuring the fidelity of the data downstream when we work with researchers to publish the data. So I want to talk about XInform, which is our name for our patient portal. And so every time a patient comes onto the platform, like I said, they fill out this short registration and e-consent, and we pull all these medical records together for these patients. We put them back onto the portal so that now patients have this personal health record that they can take around. They can share with whoever they want. If they get a second opinion, they can share it with them. We also structure this data into a care summary for them. And so as we're annotating all this data in our database, we're pushing that back to the portal so patients can see a clear, clean, graphical summary of their care. That's also very helpful to physicians when they're doing tumor boards, for example. I think if you heard Lisa speak the other day, we're working together with my DIPG Navigator to help pull this stuff together for patients that are going through the tumor board with them. And so what we can also do is offer these treatment options, basically providing clinical evidence for prior approval forms if there's off-label use of therapies, for example. But again, the tumor board is really your best resource specifically for DIPG, DMG. And the last thing that we do is, like I mentioned, we run a number of clinical studies. I'm going to talk about a specific observational study in DIPG, DMG in just a minute. And so I want to walk you through a demo of sort of what this looks like so you can get accustomed to it. So let me switch over, and this should work. So this is a totally de-identified patient case. There's a name on there, but it's a false name. And so this is what the dashboard would look like as we have the portal right now. So your care summary, you can click into it. This is going to show information about your health over time. If I'll just sort of cut over here to the timeline view, this is what that sort of simplified summary is for a past cancer treatment. And it's interactive. You can hover over it. You can see different genetic testing results. You can show or hide different aspects. And so this is a really quick snapshot that a physician can look at and get a really good sense of what was your past treatments. There's more specific information on these other tabs that go into dosing and timing, location, that kind of thing. And the biomarkers are also listed out in long form here, which can really help, again, help yourself, help physicians, second opinions to understand about your treatment. We also pull together all the medical records. This is just a quick example of some of the data that might be uploaded. If I were to click on this, we'll have the data that's pulled together. This is all obviously censored, but this is, the type of data that we can return back. And like I mentioned, we can also offer treatment options to display. There we go. It'll say a little bit about the report, about how it was generated, and these are going to have short rationales that you could use for, you could share with your physician for prior approval forms or for them to learn more about these different treatments. And so the real unique aspect of our platform is every patient that comes onto the platform, they contribute their data to the protocol, the research protocol. We can then use those experiences of everyone else to inform the next patients that come along. So every patient that participates, we have essentially your features from your cancer, and we can match that against other patients in our database, both prospectively as assisting patients in their treatment, but also for research purposes, so we can learn more. So I'm going to flip back to my presentation, and there's this slide, which I wasn't able to show because that wasn't a real patient, but in this case, this is an example of the depth of medical records that we're pulling back. And so you can see this is hundreds of medical records for an individual patient. There's an integrated viewer that from what we get from the electronic feeds, and there's just a diversity of all these different types of locations. So hospital visits, infusion summaries, orders, office visits. This is a quick view of the interactive viewer that we have. I'm going to skip through these. The last thing I want to talk about is a observational study that we are launching for DIPG and DMG. And the reasoning for this is there's really no clear natural history for the disease. And our ultimate goal is to bring an end or bring a massive reduction in the need for randomization for pediatric clinical trials. And so in our mechanism to create this regulatory grade data, we're going to create this natural history study that can be used as a benchmark for future clinical trials or as a direct comparison for external control arms for interventional studies. Again, the goal is when there's these promising treatments for patients, we want to be able to have everyone receive that promising treatment and not have to be randomized to a placebo or something that we know is ineffective. So we're pulling all this data together as a part of this study, and through a collaboration with DARPA, we're going to give real-time open access to this data to any academic or government researcher. And so in creating this regulatory grade data, we're hoping to expedite research in real time. And this over here is just a cartoon that I put together to kind of show sort of the flow and how it operates. But basically, any patient can consent through this. Legally authorized representatives, their parents, also parents of deceased patients can consent on their behalf, so we can learn from the experiences of patients retrospectively, as well as patients in the fight prospectively. And so there's just one consent. The great thing about Excelsior, our master observational protocol, is it's a master protocol. So this is just a sub-study underneath Excelsior, and there's just one consent, and we can go and collect all of this data. As part of this study, we ask for a couple different things, which is patient-reported outcome surveys, questionnaires every couple months. And we hope that this is going to be a minimal burden, but it'll also give us additional information on the clinical trajectory of the disease, as well as caregiver burden. So again, this is, we're extremely excited, extremely privileged to be able to participate in this. And we're really hoping that in running this study, we can create this really high quality data set that can be both shared with academic researchers, with government researchers, and can support ongoing clinical studies to expedite research in DIPG and DMG. This is just a quick screenshot of the data that we've collected to date. So around 150 patients. You see the records are almost 2,000 records per patient. I think to date we've gathered around 500 plus DICOM images as well. So we're pulling all the imaging together. And these are, again, just sort of rough numbers across all this. So almost 2,000 different locations where we're pulling this data from. This is my last slide. It's just a quick example of what we've been able to do. I presented this data at SNO last year. And it was just focusing specifically on ONC201, patients who were exposed or not exposed to ONC201. And we saw a large signal there, as Carl Koshman has demonstrated before. So I want to say thank you very much to all the patients and advocates, parents and caregivers. And this is our sort of hashtag for this project, which is Together We Can. I think the advocacy in this community, in the pediatric brain cancer community, is the strongest advocacy we've seen in any cancer type across ages, across the country. So you all are extremely empowered in what you're doing and what you're able to do for the research community. So thank you. Did we submit it yesterday? So we submitted the protocol yesterday. It should be open next week. Anyone, anyone that participates in Excelsior, so anyone that is consented to the platform is already participating. It's just a sub-study protocol for us to follow specific data annotation so that we want to enroll 400 patients over the next 2 years. Mm-hmm.
Okay, I had a few questions. The first question was, what level of data are you pulling from the hospitals? I know, for example, you're saying there's like 1,000 pieces of data that you get. I can imagine that there's way more than thousands of data on my son. For example, when he goes in for like his MRI, the person who does the MRI makes an inference, and then the first doctor looks at it, makes an inference, and then they have a little meeting, and there's notes that come out of that. And then, and also, are those MRI images, are the videos uploaded? Is that accessible to us? And then I have a second question, but maybe I can wait for the answer on this first.
As I'm sure you're aware, all of these different types of data have different mechanisms to collect them. We've automated most of the actual textual data in pulling that. We can actually pull most of it in 5 to 15 minutes after a request. So that's the actual raw text and export information. For actual images, we have to go directly to the radiology departments. But we get those usually within a week to 3 weeks. With this consent, it's a broad consent, and so we basically have the capability to request anything that you would request. And so, we've gotten EKG reads. It's really just being very specific about what needs to be collected and what needs to be brought in. For this specifically, for this program, it's focused on clinical data, raw imaging data, and next generation sequencing, genomics data.
Okay, and my follow-up question is, receiving all this type of data, I've worked on a lot of data projects, and I know that the data doesn't come in clean, especially across different institutions. And I know that, I work in corporate America, and it has taken a lot of manpower to sort of clean the data in order to make it usable. How are you guys cleaning the data, or choosing, like I was saying, that first MRI inference that comes from that radiology person, and I know you're saying you're not collecting that now, but there may be other examples of how like that might not be as useful as the last inference on that same MRI that's had doctors talk about it. Like, how do you distinguish if you have a computer coming in that's looking for a particular word, in order to, if you're doing it in 15 minutes or whatever, then it's got to be done by a computer. Like, how do you do that?
So you nailed it. We actually have multiple levels. So the first level is we pull and sweep absolutely everything. And so that's an electronic pull. We have some natural language and named entity recognition algorithms that basically help us to understand it. But at the end, the actual research grade data set that we're offering access to, that's all manually annotated by our team of data annotators. And so, again, it's analogous to how CPTN is doing their work, where they bring in the records, they have a group of annotators that annotate all of the data according to a standard data model. I kind of skipped over it, but we normalize all of these raw terms to biomedical ontologies. And so we can normalize all the raw text to standardized entities. Thank you. Good. Thank you, everyone, again.
Thank you, Tim. For those of you who have not yet visited the DIPG 1 link, please do. We also have links to Xinform and other information regarding how to share data in there. For our next demo, we have Peter Gilbertson as a technical project manager with Leaders Biomedical Research supporting the National Cancer Initiative, CCDI, an initiative seeking to build a community centered around childhood cancer care and research. He has spent over a decade with Health and Human Services, advancing research and technology projects in public health and healthcare, and currently works on the development of tools and processes to promote the use and understanding of pediatric cancer data. As part of the CCDI team, he led the development of CCDI Hub and the Childhood Cancer Data Catalog. Both projects seek to share pediatric cancer data and resources with the members of pediatric cancer community and enhancing linkages to pediatric cancer resources. Peter will be joining us remotely, so welcome.
Thank you very much. I appreciate that you guys are letting me join remotely today. Although I work for the Frederick National Lab supporting the NCI, I am based in South Dakota. So it is extremely kind that you're allowing me to talk about two projects that I've worked a lot on. And so I appreciate that. So today I want to talk a little bit about the CCDI Hub and then another CCDI developed project, the Childhood Cancer Data Catalog. So I'll just give a quick overview of the two applications, the hub and the catalog, and then I'll move over into a demo. And before I do the demo, I'll just give a quick use case so you can see how we're going to use the catalog. And then I'll put up a few helpful resource links. So if you're interested in learning more about the hub or about the catalog, you can follow those links out there. So first, what is the CCDI Hub? So as was mentioned, this is part of the Childhood Cancer Data Initiative. So this project is seeking to build that community around pediatric cancer data and seeking to sort of build the tools or build the relationships for connecting data, analyzing that data, and ultimately sharing that data. And what the hub is the hub is meant to be the single location, sort of the one-stop shop, if you will, where people can come and learn about applications and tools that have been developed as part of the CCDI to sort of further that goal of linking, sharing, and understanding data. The idea behind the hub is that CCDI has supported a number of different applications. You can come to the hub, you can quickly learn about those applications, assess whether it's related to the work that you're doing or related to the area that you're hoping to explore further, and then navigate directly to those applications. So it's sort of, I like to say it's a site designed for people to leave, where you come to the hub, you identify the resource of interest, and then you immediately navigate away to that resource of interest. One of those resources of interest that I'll be talking about today is the Childhood Cancer Data Catalog. And so I think this catalog is sort of building off of some of the topics that have been discussed earlier, certainly in the morning session, about pediatric cancer data. And while the hub is not at an individual level, the hub is more, or not, sorry, the catalog is more at a resource level or a data set level. So the aim of the CCDC, or the data catalog, is to inventory different pediatric cancer resources, be those resources repositories, registries, data commons, biospecimen repositories, different types of resources that have different tools or different data sets about pediatric cancer resources. The catalog catalogs them and provides some high-level summary information that allows you to assess what is available in that resource or is available in that data set. Again, make some quick, informed decisions. And then once you've identified whether it's a resource or whether it's a data set or something else, that one of these areas is relevant, that you can then navigate from the catalog to that area of interest. And so that, sort of each step of the way, from the hub to the catalog to the resource itself, you're sort of building layers of information, where the hub has just the briefest, the catalog we hope to add a little bit more, and then you navigate to that individual resource or that data set where you can either access or understand or come to greater awareness of what's in that resource. So as it kind of says here at the bottom of the slide, like, the goal of the catalog is to make it easier for researchers or truly anyone to find information about a particular pediatric cancer, and then navigate to that resource or navigate to that data set. Just a kind of summary of what is in the catalog. So the catalog we have, so the slide says 250, but we actually just had a release on Wednesday. So we now have 253 different pediatric data sets belonging to 53 different resources. And so these data sets and these resources cover, again, a wide range of topics, from biospecimens to epidemiologic data to imaging and xenograph. There are not limits to the things we are trying to catalog. The only limit is that it needs to be related to pediatric cancer, or it needs to support the analysis and understanding of pediatric cancer. And then one thing special about the catalog is that the catalog currently is the repository for making connections out to CCDI data. So I mentioned that the hub is the tool or the application that connects to the resources developed by CCDI. CCDI also supports a number of research studies and projects And the catalog is the location that you can learn more about those projects and then connect out to those individual projects. So next I will do a demo. Before I do that demo, I'll just kind of talk about, a use case. So the demo, I'll start with the hub, kind of showcase the hub, and then we'll navigate to the catalog. And then I'll sort of highlight using the catalog to search for information about glioma, leveraging our search catalog feature, our ability to search through the data sets, highlighting the information within each data set, and then sort of talk about the next step from our data sets to the resources themselves. Give me a second to switch over. Here, let me share slightly differently. So I have pulled up here, this is the CCDI hub. This is the live site that's out there, ccdi.cancer.gov. And again, this site is intended to be one location that you bookmark or the one location that you're aware of if you're looking to connect with the applications and tools developed by the Childhood Cancer Data Initiative. And again, the idea behind the site is to list the tools, briefly describe the tools, and then encourage users to navigate to those tools and leverage those tools themselves. So the site is really set up in order to highlight those tools. We have our carousel where we are highlighting the individual tools listed. Our menu bar, again, is listing all of the tools available through the hub and supported by CCDI. And then the homepage itself is sort of highlighting information about those tools. So we have just some general stats at a glance of some of the counts and information within the tools that are supported. We have latest updates. So as different tools are making changes and updates, we try to centralize those updates on the hub so that if one particular tool has changed in a way, you can come here and see what changes there are. And then we have just a listing of all of the tools. So we have CCDI supported tools and applications, and then we have other resources as well. So CGC and then dbGaP. These resources are sort of supporting CCDI as well. So whether it's data or something like that, that will start in the catalog or start in the National Childhood Cancer Registry Explorer, and then maybe access or information is additionally stored in either CGC or dbGaP. So again, the idea is these short, sweet descriptions of the tools themselves, and then let's navigate away from the hub to the tool. So I'll navigate to the data catalog. And now we have left the hub and we have moved on to the data catalog. So again, the highest level is the hub. There's a little more detail in the data catalog. You'll see here on the data catalog, we have some information about the catalog itself, and also about the resources that are there. So we have the search tool, the search catalog, we have a listing of our participating resources. And then as I mentioned, we have information about CCDI studies, CCDI data sets, and then also information about accessing that data. On the homepage, we have just general information about the catalog. The catalog itself tends to update fairly frequently. Every month or so, we like to update our data sets so that the data in the catalog reflects the data that's available at our resources. We have a listing of data sets, and then generally we try to navigate people to search those data sets to find the information they're looking for. So in this case, as I mentioned, we'll use our catalog search to look for glioma. And so the way that the catalog is set up is that we have, as I mentioned, 253 data sets, and the catalog search allows you to do a free text search of those data sets. So in this case, we are searching glioma, we are searching different disease diagnoses, and we're getting back a list of all of the data sets. So as I mentioned, we started at 253, and we're now at 51. So 51 data sets within the catalog touch on glioma. As I mentioned, we have 53 resources. So you'll see here, you can also filter by resources. So as you make text searches, the resource columns will filter as well. So for glioma, you can see that the resources, CCDI, the Childhood Cancer Repository, dbGaP, all of these resources have information about glioma. And then you can sort of filter further down if you're interested. So if you only wanted to see CCDI, we can now filter by glioma and CCDI and get just 4 results returned. For each of the data set, we have done some initial digesting. Again, the idea is to get some high-level summary information to let the interested party know what information is there. If there's someone interested in doing research, case counts, sample counts, so that you can assess whether there's enough information to do the type of analysis you're looking for, or just information about the description and the types of diseases for those interested in connecting more with those resources. So you'll see here for CCDI, for the comprehensive genomic sequencing of pediatric cancer, we have these different diagnosis types. We have 194 cases. We have 373 samples from different assay methods, and then a general description of it. For each data set, you can click on the data sets themselves, and you get more information. So now we have information on, again, the resource it belongs to, the point of contact for this resource, the description of this resource, and then we have more breakdown of information about the data within this resource. So in this case, again, we had those 194 cases. We can see the breakdown by gender, male, female, not reported, information on race and ethnicity, and then more complete information on the different disease diagnosis values, assay methods, and then sample anatomic sites. For each of these items that we are calling our core data elements, so number, sex, age, race, all those can also be visualized. So we have charts set up for each of those. So in this case, we have sex, we have male, we have female, but we could change that to be disease diagnosis. And then you can see a breakdown of this information visually, which makes it a little more easy to digest. Beyond just this count information, again, the idea is that we are connecting you with a data set or with a resource. So we have links out in this case to dbGaP. So you can click on this link, then you can navigate directly to dbGaP. The other thing you can do is, if this is of interest and if there's interest in connecting with CCDI, again, each of our data sets are categorized under a particular resource. We can navigate to that resource page, and we can see more about that resource in general. So here we have the CCDI resource, again, a description, what type of resource it is, specialization, the data that's available from this resource, when was it last updated, the types of data that's available, and then also how to get data access. So in this case, CGC, the data from this resource is stored in CGC. We have that data access link that you can then navigate away. And so the idea is that you'll come to the site, you'll do these searches, you'll find data of interest, and then you can navigate away to either request that data or leverage that tool or view those images. But you can come to this single resource to do that initial assessment at a high level that you can then go out and explore or connect or build that sort of community of data sharing that the CCDI is supporting. The other thing I'll note is we do allow filtering on different resource types. So we have a lot of different resources. We have analytic tool resources, biorepositories, knowledge base, registries, and then we have lots of different types of data that we share within the catalog. So we have biospecimen data, clinical data, imaging, xenograph data. We have lots of different data that we try to capture here, and all of it relates back to pediatric cancer. I'll switch back over to my slides. And so generally speaking, that's sort of the workflow that we envision for both the hub and the catalog. The hub is the tool that connects to the catalog as well as the other resources developed by CCDI. And CCDI is that tool, hopefully, that can help connect researchers or community members to the different data sets or to the different resources working on a particular cancer. That said, we are always looking to grow, certainly the catalog itself. we are looking to grow the information that is there, adding more resources, adding more data sets, sort of, again, building up what is available to search and what information we can make available to the user community. And then I'll just end here on this slide. Again, the hub is ccdi.cancer.gov, and that's really the entry point that allows access to the different CCDI tools. And so you can go there and you can see what's available, and then you can explore the catalog some more, or you could explore the Molecular Targets platform, another tool we support as well. But the hub is kind of the starting point that then sort of opens the door to CCDI applications and projects. And that is all I have. I don't know if there are questions. Being remote, it's harder to see.
Are there any questions? Another handful of people here. That's it. Thank you, Peter. Thanks for the presentation. I appreciate it.
Yeah. Thank you for having me.
Good morning or afternoon, everyone. I'm going to hold the mic, and I'm going to be asking for my support team here to let me know if I'm speaking too loudly or too softly. And I'd like to introduce myself. My name is Marilyn Dyson, and I come from a background of mental health therapy for 15 years. I started off working with victims of domestic violence in Los Angeles. I serviced nonprofit organizations, and I serviced individual therapy. I've serviced individuals who needed therapy with either depression, emotional disorders, and so I'm here now. I just want to self-disclose. I'm not practicing anymore as a therapist. But I do work for Accenture, and one of the things that I do at Accenture is work with our teams to create psychological safe environments in the workplace. And so my background really, in therapy, mostly I worked with children, and so Defina has asked me to come here and share coping strategies with you all, but I really want to make this as authentic as possible and hear from you and allow you to ask questions and allow you to share your stories. And so I'd like to start by saying, by asking all of you to just say your child's name out loud and say it 3 times so we can all hear it. On the count of 3, please. Everyone say your child's name. 1, 2, 3. Thank you. I'd like to continue by saying that it takes a lot of courage for you all to be here today. And I want to take a moment to let you know that I empathize with each and every one of you. I am a mother of 2 children, and I honestly cannot personally imagine what it is like to experience what you all have experienced. And so I just, from the bottom of my heart, want to offer my condolences to all of you. So today, I also like to I'm sure a lot of you have heard of various coping strategies. Some of you might have been in therapy. Some of you have been in therapy for X amount of years and still feel like it's a bottomless pit. And you know, we all grieve in different ways. And the one thing I want to say to everyone is to please be patient with yourself. Please be patient with yourself and understand that others in your life don't always necessarily know how to support you. They don't know what to say. And sometimes they say some of the most craziest things. And so as much as you can, try to have patience with them as well, okay? Because most of our loved ones want to do the best for us. They want to do things for us, and sometimes it's just not working. So keep in mind that a lot of times they've never been in your shoes, and they really just want to love and support you all. So so again, I'm going to share some coping strategies, and I'm going to ask if people would be willing to share what they have done. Because a lot of times, if you've ever been in support groups, and actually, let me ask, has anyone ever been a part of a support group? Please raise your hand. Wonderful, wonderful, okay? So in support groups, a lot of times it works for those to hear other people's stories and to hear how other people have coped, because we're still reaching for answers, we're reaching for ways to stop feeling a certain way. And so sometimes support groups are very helpful. So I'll just go there and say a coping strategy could be going to a support group that specifically deals with these issues. And so please utilize those groups. connect with people that are in those groups. And also sometimes on your stronger days, you can be a stronger leg for others in those support groups as well. And so I'm going to ask a couple of you if you're willing to share your story in brief in here with us today. And also, we're going to have general Q&A. So we don't have a whole lot of time, but for those who are tuning in live streaming, they're going to post my phone number. And for those of you who have questions or you want to share your story, I have my phone here, and we're going to do it a little bit. You know, I'm not technology forward like people really want me to be. But if you would like to text me a question or share your story of your son or your daughter, I welcome you to do that. And so you can start doing that now if you like through this presentation. So coping strategies, again, some of these strategies you probably heard before, but maybe in a different way. And it's never too many coping strategies to hear about and learn about. So one of the things that I'd like to, another thing I would like to suggest is to share cherished memories. It's always okay to share cherished memories. It's healing to find a safe place and talk about your child. How did they play? What were the things that they used to do that made you laugh? What did other people say about your son, your daughter, your brother, your sister? To memorialize. And also, has anyone ever heard of planting a seed or planting a new plant in their yard or something like that? That's another beautiful way to memorialize your child. Talk about your child's emotions. Talk about what made them hurt, what made them laugh. What stories bring you joy? What stories bring other people in your family joy? Creating meaningful rituals. Of course, celebrating their birthdays, celebrating that amazing day, first day of school when they first met their kindergarten teacher or their preschool teacher. What day was that and what did it look like? Do any of you keep journals? Yeah, sometimes journaling helps a lot too, to memorialize your child. And also art. There is such thing as mindful art, where sometimes it's guided and sometimes it's not guided, but just being very still and writing shapes, drawing a circle over and over and over again, and just being in that space of drawing circles. So art therapy is also very, very soothing, and it's very healing as well. And self-care practices. I see more moms in here. I would assume that it's mostly moms. And I'm not sure who's streaming in, but I really want to remind you, moms especially, to take care of yourself. A lot of times as moms, we do self-neglect because we're taking care of everybody. And especially if we are mourning, we forget to drink our water, we forget to exercise, we forget to love other people. So let's not forget to take care of ourselves. So also finding trusted friends and close friends who are patient with you, who would be happy to listen about what you're feeling that day, what you have been feeling for weeks, for months, for years. And I know I said it before, but I'll say it again. Please be patient with yourself. Grief it comes in ebbs and flows. The emotions of grief comes in ebbs and flows. Some days you might feel fantastic and you're thinking, this is great. Sometimes you may even feel guilty for having a happy day. It's okay. It's okay. And then if you want to spend that day crying, if you want to spend that day yelling, cussing, screaming, it's okay. Give yourself grace. Because sometimes not everybody is going to give you grace. Give yourself grace. Be patient with yourself. So I'm going to go ahead and ask the room, ask those who are tuning in, what have you done? What have you done? And please, if you don't mind, sharing with us how long you have been experiencing the feelings that you're dealing with today, and help us learn. What have you done? Anyone who wants to volunteer, please raise your hand, and I'd like to create a safe space for all of us here.
After my son, Dylan, passed away, he was 17 when he was diagnosed with a highly aggressive pineal tumor. He underwent 3 surgeries, 3.5-year battle. He was extremely athletic prior to this, an honor student. He was extremely bold. He had a great sense of humor. He could light up a room and everything, but he fought. He fought until his dying day, right away after he passed, and that was in August 2021. He was 20 years old. I knew I needed to reach out to people that would understand what I was going through, because this was a very tragic type of death. All children that pass, it's tragic. But what these children go through, no human being on this earth should ever have to go through. So I reached out to the Pediatric Brain Tumor Foundation, and I said, I was wondering if you have a bereavement group, and they said, for the first time ever, we're starting one in 2 weeks. And I thought that was a miracle, because I felt like I was on another planet. I was very isolated, and I got into that bereavement group, and right now I'm still in that group, but they have already have now 7 different groups. It's growing, and they didn't even know if it was going to continue. It started once every 2 weeks. We would meet for 4 months, and then once a month, and they didn't know if this would go on, but anyway, it's still going strong, and it's been a little over 2 years. So that's helped me a lot, and that's why I wanted to share it with everybody here, because every day is it's a challenge to get through. Thank you.
Thank you very much for sharing your story. And for those who may have missed it, the Pediatric Brain Foundation is I'm sorry? Pediatric Brain Tumor Foundation. Brain Tumor, I missed that word. Pediatric Brain Tumor Foundation. Anyone else? Thank you again.
My name is Roger Ward. My son was Jace Ward. I still can't really say his name without crying. Sorry, guys. It's been 2 years, and miss him every day. But right away, after Jace passed away, we got invited to a bereavement seminar down in Florida, and we went. I did not want to go. Guys, men in the room understand this. Men grieve differently than women.
Sure.
I just wanted to be alone. Didn't want anybody around me. My wife, on the other hand, she needed contacts. She needed these lovely ladies right over here and everybody in the room. But not me. Didn't need anything. Couldn't talk about it. Didn't want to talk about it. But I went, and it's amazing because as men, we don't talk about it. And it was great because all the guys there was the same way. We told wonderful stories about our kids, and we didn't have to talk about it, but we bonded because we all went through the same thing. And to this day, they're my brothers. I'll be there for any of them, and they'll be there for me. But I think that the biggest thing that I want to put out there for any of the guys that are grieving, and I know I have a brother over here that's recently lost a wonderful daughter, but find people that shared it, because you don't have to talk about it, but you just know. It's easier that way. And that's different for men, but that's the way we look at it, I think. The way I did. And, you know, I have wonderful friends out of this too that just really helped out. So that's my story.
Thank you, Roger.
Hi, my name is Sylvia. My son was Noah, and I've been in this horrible life for 3 years. And I'll piggyback off of Roger over there. I was at the same retreat. I wanted to go because that's how girls grieve. I needed these ladies. My husband did not want to go. And having a couple grieve so differently all the time is extremely stressful on the unit. I don't like to sit in my grief. I visit my grief, and then I come out. Because if I stay in my grief, I will not make it. And I know that. My husband likes to live in his grief. And so that right there was really hard for us. Turning point for us was 1 year in. I lost Noah on July 12th. Jace left us on the 3rd of July. When I got the news, I waited for my husband to get home from school, and I said I packed up everything. I drove overnight to Kansas to be with Lisa and Roger, and Antonio had some really, just some moments that were absolutely what turned him around and wanted him to grieve a little bit differently. I think I was with Stacy. We went out for breakfast, and we were in a room all by ourselves. Nobody was there. We had a great breakfast, about 2 hours. We got up to leave, and Todd, Stacy's husband, saw something on the floor and bent down and picked it up, and it was a Luigi. Luigi was my son's favorite thing. The last thing Noah and Antonio did together was play chess. And Noah loved the queen because he said that she gets to move wherever she wants. And the Luigi was a queen. And it was under his chair. And, Antonio was it was the look on his face. And Todd's like, does this mean anything to you? And Antonio was like, yeah, we still have it. It's on his nightstand. On our way home, on our drive, I said, I think your son's trying to tell you something and you're not listening. And at that moment, a green Jeep cut us off, braked really hard. He had to brake. We flew forward. And then I turned around, I looked at him, I said, you're not listening. You need to listen. And that was the turning point for us. We decided that we need to grieve differently. And so I know he now has a lovely group of gentlemen that are just great friends. And I have a wonderful group of ladies. I don't always grieve right or correctly, or I don't know if you guys, I don't sit in my grief. So a lot of the time I get, you are doing wonderful, but they just don't see me when I walk into my grief. My grief is there, just like every other mom. There's no right way to grieve, so you just, I guess, have to find what works for you. And this is all I know right now, so, I'm sure it'll evolve because this is the life I've been dealt with, and I'll be here forever until I'm no longer here, so.
Thank you. Did you say Sylvia? Nice to meet you, Sylvia. Thank you for sharing your story about Noah. And Roger, thank you for sharing about Jace. Dawn, I didn't get your son's name? Dylan. Dylan. D-Y-L-A-N. Thank you. Sylvia, you said you just sit in your grief?
Sometimes.
Mm-hmm. Do you feel bad about that?
Well, my therapist says that I do a lot of avoiding and I do a lot of staying busy to not sit in it long. So we're working on it.
Yes. And I would hope that some of you or all of you would know that it will probably never go away, the feeling of grief, okay? Sometimes people have expectations that we'll just get over it. You might even hear someone say, well, just get over it. And even though they don't mean to be hurtful, we'll never get over it. We'll never get over it. But each day, you take one day, one step at a time. And if at the end of that day, you can think about something great that happened, or if you find a Luigi, that was a great day.
It's a great day.
So look for those little signs, because your son, your daughter, your sister, your brother, your niece, grandchild, they're still here. Their spirit is still alive. So their body, of course, is not here in the physical. And I understand this is a painful process for them to go through. Medication and things like that. Is that, oh God.
It's a short and brutal disease.
It's like ALS for kids. They know everything they're going
They
through.
stay physically, mentally sound, but they lose. Noah could play chess the day before. He had very limited mobility, but he knew, I can't remember where pieces go. Like, I can't play chess, I'm sorry, guys.
Sure.
But, Noah was just, he was playing, and he would move it a pawn, and Antonio would be like, well, I'm going there, and he's like, no, like, you're not going there. Like, you can't move there.
Personality.
So they stay
present,
but they're conscious of all the things that they've lost, and that's a very hard thing. I think my friend here said it best. I think you made a post a few years ago about watching all of Hudson's lasts like they were his first, and it gets me every time.
Thank you for being there for each other. It means a lot. Is there anyone else who would like to share their story? I haven't received any text yet, so those who are here live streaming, I do not want you to feel left out. Please reach out to me and share your story, or even a coping strategy that you might want to share with us today. Anyone live in this room would like to share?
Hi, I'm Linda. My son is Charlie, and as far as coping strategies, he died 5 years ago, and I think I agree with all of you in that being around others who understand and being able to, I don't have to explain it. With my friends who have not lost children, they kind of make me explain, well, why? I'm like, like I don't, if I have to explain it, it's too hard to even explain. But we can finish each other's sentences. Oh, I know, I understand. And that is, I think, one of the reasons I come back to CareFest every year is just to be around others who get it, and we all support each other, and I can listen to them about their child, and they ask about my child, and I can say his name. And I think that is the best. And I can meet people who have been on this path longer, and they give me hope because they're still here. Sure. And then I can look at others who maybe are newer in this and hopefully, share some things as well. I mean, I don't like to give advice, but just to say, he's not forgotten. I carry him with me, and I now can remember the good things and memories, which is I'll take that.
Sure.
So if I haven't met you, I'd love to hear your kid's name, and thank you for sharing and listening too.
Thank you, Linda. Thank you for sharing about Charlie. Anybody else?
I didn't want to take the time away from anyone who hasn't been able to share, but my daughter's name was Tatum, or is Tatum, and she'll always be with me. She was 4 when she passed from DIPG, which is the tumor on the brain that is with death sentence still. So I'm a nurse, went into nursing to take care of all my kids, be able to take care of them, whatever happens. This was supposed to be an ear infection that just went awry, so it wasn't an ear infection, it was a brain tumor, the problem was. So we just had to get the advice to take her home and enjoy her and make memories. And I'm sorry for the newly your loss. I haven't met you yet. Some of you may know me from the bereavement tent at CareFest. We've been doing this for a few years. We like to have the parents or siblings put the name on a flag, and we have the names from the past, and we put them up every year, and we'd be more than happy to see you over there at the bereavement tent, and go ahead and we'll get your child's name up there, and we welcome you. I'm not licensed in any bereavement counseling or anything, but we do have bereavement sessions, and the dads get together, the moms get together. It is very important to get together, share your stories, say your child's name. We do the say your name out there on the stage later on, and it is powerful. It is, I get a lot from it, just offering the time to come to Washington and be part of the bereavement, because when we first got here the first year, it was just a wood table with a very nice young lady behind it. She wasn't a sibling, and she wasn't she didn't have anyone pass, but she was running the bereavement table, and there was nothing there, so we just decided to kind of step up into that void. And we've met fabulous people, and coping wise, well, I haven't met my therapist yet. I don't have a therapist yet, but except for my friends. But yeah, so a lot of times I'll tell dads that I think I'm doing it wrong, because I just to provide for the family and be there for my wife and children, I kind of avoid. I kind of pushed off things. I prayed for numbness, and sometimes it hits me really hard, and I'm sure there's someday I might just lose it, but we all do things differently. Yeah. And I'm sure my therapist, that's not my therapist yet, would tell me avoidance is terrible. So, so that's my piece for today, but I want to just invite everyone to come over to the bereavement tent if they get time, and be happy to get to know you and love to hear about your kids.
Tatum's dad. What is your name?
Scott.
Scott. Nice to meet you, Scott. Thank you for sharing. And again, the bereavement table. Please visit Scott. Yeah. Thank you for sharing, sir.
Jim, my daughter's Esther, for such a time as this. What helps me?
Try your best. Amen.
Thank you.
That was the best video I saw today. I got to see it because I'm sitting up here. I saw the angle. That was awesome.
Anyone else? Earlier, I spoke to the moms because I am a mom, but I also like to speak to the dads. Roger saying you don't like to talk about it. That's perfectly okay. Men grieve differently than men or than women, but women amongst women grieve differently too. Everybody grieves differently. It's ebbs and flows, happy days, sad days, angry days. These feelings are okay. Be in those feelings, okay? And I love, Tatum, the way that you corrected yourself when you said was. Tatum was, she is. She still is. So at this time, if there is no one else that would like to share, I don't have any text yet.
Sorry,
please, I'm
kinda
sorry.
No, you're okay. My name's Melissa Moore. My daughter is Madison Moore. She went by Maddie. She passed away 3 years ago from DMG. I just wanted to share, when you said you were told to go home and make memories, you know, we kind of got the same thing. One of her hardest, you know, one of the hardest things that she had to deal with her tumor is she lost her short-term memory. She never gained that. Gosh, so sorry.
Please don't be with it.
She was never able to make memories again. So each day truly was a new day for her. So towards the end of her battle, her doctor put us in a room and had Maddie go with our nurse and kind of separate us. And she was giving us that talk that, you know, it's getting close. And she just shared with me this advice and that I've always kept close to me, especially during my grieving period. And she said, go home and just live in the moment, because Maddie truly just lives in the moment. She doesn't talk about tomorrow. She doesn't ask any questions about tomorrow. She never asked any questions about yesterday. She truly just lived in the moment. So anyway, to this day, when I find myself having a really hard time, or, you know, in my grief, I'm in my hole is what I call it. I just think, I just need to live in the moment. I need to go on a walk. I need to, get fresh air. I need to, see a bird or something. And I just, live in that moment. And that kind of just helps me take away the thought of, I have all these years ahead of me that I won't get to see her. But it kind of, brings me back to a grounding place of just living in the moment that she truly did. So, that's kind of my little message.
That's beautiful. Thank you for sharing about Maddie. There's someone. Oh, she doesn't need the microphone.
Well, the people online.
Oh, okay. I don't really know what I'm going to say, except that I feel like I need to say his name. And his name is Mikey.
Mikey.
And I can't even say. He was 19. This is him. Big athlete, just a big bear. Just loved everybody. And everybody loved him. And it just isn't fair. And I miss him. All I can say.
Thank you so much. You know, just the little bit that Mikey's mom just shared is super, super powerful. She's one step ahead from where she just was before she said something. Baby steps, a day at a time, a breath at a time. All of you are here, and I'm just amazed. I'm really, really, really touched by seeing you here because you got up this morning, you put each pant leg on one at a time, and you got here. You all got here. Everyone is dressed, and you're breathing, you're sitting up straight in your chairs. You did that. So even if it's those really little things that might seem very simple, very silly, you did that. And there are so many other things that we can do for self-care, and like I said earlier, I relate as a mom, but dads, take care of yourself too. Take care of yourself. A lot of times dads, want to step in the role of, the protector, taking care of all the rest of our children, taking care of the family. But dads, you guys deserve your time too. You deserve your time and your space too. So please make sure you take care of yourselves as well. Exercise, folks, all of us need to be doing that. But find something that works for you, that you can do. Whether it's an art class that you join where you have to get there at a certain time to, make sure you get your favorite seat, or a Pilates class to make sure you see your favorite Pilates instructor. Whatever it is, do it. And praise yourself for it. Pat yourself on the back for that. Love yourselves through this. In the mornings, sometimes we're not even sleeping, and I want to recognize that as well. I'm not a medical doctor, obviously, but if you are having trouble sleeping, if you're having health issues behind this, please make sure you're seeing someone to help you through this process, whether it is a therapist in conjunction with a physical doctor. Be very, very open about how you're feeling and what's happening physically to you, or what kinds of thoughts you're having. There are lots and lots of thoughts that keep us it can keep us in a very stagnant state. These emotions, these symptoms of depression could be a clinical depression. It can be temporary, it could be acute, or it could be long-term. But regardless, we all should be seeing someone if you're not seeing someone. It's imperative. You're here living for a reason. Continue to stay on that journey to figure out what that is. So I don't have a lot of time, but do I have anyone else who would like to, please? Yes.
Hi, I'm Angie. I'm Sarah's mom. Sarah was a little bit older when she was diagnosed. She was 14, and she fought brain cancer. She fought a germinoma. At 18, she relapsed, and she was 18 years and 6 months and 11 days old when she died. She spent the last 10 weeks of her life on life support. She was intubated September 24th of 2018, and she died on Thanksgiving Day of 2018, which has caused a lot of issues for our family as far as celebrating Thanksgiving. There's some people who want to pretend like that didn't happen on a holiday, and then there's some people who don't want anything to do with the holiday at all. Because she died 5 years ago, her friends are now getting married. Some of them are having babies. So that's an additional thing where I'm like watching her friends go on with their lives and do all of these things that she didn't get to do. And she was kind of on that cusp of adulthood, but because she had cancer at 14, she was kind of held back from that a little bit. So she wasn't, a typical 18-year-old. And of course, at the end, she was just, intubated. She was put on ECMO. The last 7 weeks of her life she spent on ECMO, which if you've ever seen anybody on ECMO, it's really brutal. While she was on ECMO, she was on blood thinners. They did a tracheostomy that was very bloody and very awful on her. She was awake for it. They couldn't get her sedated enough, and they just went ahead and did it. I came in afterwards, and she had dried blood in her eyebrows. Like, the horrors of having a child on life support for that long, it's just unimaginable. And
Angie, we have 1 more minute and this is particularly for the live streaming.
Okay.
Do you mind if we continue to have a discussion?
Absolutely.
I'd like to thank everyone for sharing your stories and your tears and your thoughts with us. I'm sure there's still a lot of fear as well. For those of you who still have questions that are live streaming, my phone is still working, but I don't have any text yet. I will stay around after this is over. So if you want to continue to speak, I will be happy to speak with you all. So again, thank you so much for being here, and I congratulate each and every one of you for being here today. Thank you.